Angelini, and I am coordinator of the PhD course Translational Specialistic Medicine, GB Morgagni, and together with Professor Gregori, we are welcoming you to this first edition of the Winter School, Shaping a World-Class University, seed for founding for digitalization and innovation. This school is meant to address one of the 17 goals of the Europe 2030, especially dedicated to quality education. And the idea has been to organize this winter school for all the PhD students of the Padova University, in particularly for those of the School of Medicine and those also of the Pillar Network that you know are a group of universities that join Padova University for education, research and exchange. The aim is to offer knowledge and competence in the field of artificial intelligence and big data in the biomedical research. And the setup of an international school is based on the contribution of prestigious international faculty with outstanding guests coming from high-ranking universities from Italy, Europe and US. This will promote and favor exchange also between our PhD students and reinforce the Pillar Network. Organized this winter school in 5 afternoons. It's a virtual, and we are recording, and some of the material will be available also later on. And each afternoon we have chosen a group of experts in the field from these different universities, and the topic are 4 different topics. The first one this afternoon will deal with controversies in statistical learning in clinical epidemiology, coordinated by Dario. The second topic will be machine learning perspective in novel field of biomedical research tomorrow. On Wednesday, we will touch on the topic of data science for omics sciences. On Thursday, we will deal with artificial intelligence in pathology, and on Friday, machine learning and prediction in COVID-19 epidemics. There will be possibility to interact, even though it's a webinar, and we will be able to pose questions through the chat. I will leave the podium to Professor Dario Gregori for the rest of the afternoon. And thank you to the speakers and the attendees for joining this course.
Thank you, Annalisa. Thank you, Professor Angelini, for this introduction. As you told us, I be on duty this afternoon by chairing this exciting session. Let me share the screen just to briefly go through the program. Okay, here is it, and It should work. Okay, this afternoon the topic will be statistical learning and clinical epidemiology. The structure will be that after a short introduction by Giulia Lorenzoni, we will have the three outstanding speakers, Frank Harrell, Ron Summers, and Rob Tibshirani, giving us their perspective on this innovative field of research. As Professor already said, you are free and welcome to interact with the speakers through the Q&A section. You see there is a button somewhere in the Zoom, and you can write down, and it will be a responsibility to share your questions with the speakers. Ideally, leaving the more complex and questions and comments to the floor discussion that will follow immediately after the last speaker, Professor Tibshirani. So I'm starting giving the floor to Giulia Lorenzoni, the postdoc fellow working in our university. She got a PhD in healthcare planning and research, and she has broad experience in doing clinical research, leading the laboratory in clinical epidemiology in my unit. So please, Giulia, start whenever.
Thank you, Dario. Do you see my screen in presentation mode?
Yeah, perfect.
Okay, perfect. Thank you so much. Good afternoon, everyone. And thank you, Professor Angelini and Professor Gregori, for the opportunity of being here today, introducing the first day topic of this school, today about controversies in statistical learning in clinical epidemiology. I would like to start my presentation with a citation from a paper that was published in JAMA in 2016, saying, "By now, it's almost old news. Big data will transform medicine. It's essential to remember, however, that data by themselves are useless. To be useful, data must be analyzed, interpreted, and acted on. Thus, it is algorithms, not data sets, that will prove transformative." I think that this sentence synthesizes very well the today topic. Trying to broaden the concept, we might say that it is the method that makes the difference. Without an appropriate methodological approach, data collection, data analysis, and data interpretation, medical data will be completely useless. And to identify the most appropriate method for evidence generation in the clinical setting is the core business of clinical epidemiology. Clinical epidemiology is an year discipline starting its development from the beginning of the 20th century, when in 1938, John Paul, he was an infectious disease internist, during his presidential address to the American Society of Clinical Epidemiology of Investigation, defined clinical epidemiology as a new basic science for preventive medicine. So at the beginning, the focus of clinical epidemiology was population instead of single patients. But things changed over the years, and in the 60s, the focus of clinical epidemiology changed from the community to the single patients or groups of patients in the clinical setting, no more in the community settings. The 60s were important for the development of clinical epidemiology because in 1968 it was published a landmark series on clinical epidemiology in the Annals of Internal medicine, where the field of clinical epidemiology was defined as the clinical statistical study of disease populations. Such an increasing connection between clinical epidemiology and clinical care allows clinical epidemiology expanding beyond its traditional focus on infectious diseases to all the broad range of diseases, from acute to chronic diseases. And thanks to such an evolution, clinical epidemiology gives an important contribution to a better understanding of diseases, risk factors, prognosis, treatment, and also it contributes to the reduction of patients' mortality in the long run. At this point, it appears clear that data analytics is one of the most critical aspects in clinical epidemiology. When I talk about data analytics, I refer to the use of both traditional statistical methods and innovative machine learning method for the analysis of the data. Traditional statistical methodologies refer to regression-based models. Some examples are represented by ordinary regression, time-to-event models, longitudinal models, and so on, that are those most widely used to analyze the clinical data in clinical epidemiology until now for the purpose of prediction or to estimate the effect of a treatment. But on the other side, in the last years, a growing interest has been developed towards the use of machine learning approaches also in the clinical epidemiology. But before talking about machine learning, I think it is very important to try to understand what we mean with machine learning, because the concept is not straightforward. Machine learning could be defined as the study of algorithms that improve throughout data experience. So, in simple words, they learn from the data. It is clear that the traditional statistical models present with several differences compared to machine learning models, and it is extremely difficult to synthesize all such differences in just a presentation. But I would like to point out one of the most important differences to me between traditional statistical models and machine learning. So that when we use traditional statistical models, we make assumptions on the data structure we used within the model. An example of such assumption is the proportionality of hazard for the Cox proportional hazard model. While on the other side, machine learning models the data without assuming a preconceived structure. Such a flexibility and lack of assumption of machine learning approaches is what made so popular machine learning in the last years. Given such differences between traditional statistical models and machine learning models, and the two different underlying philosophies, they have been proposed for a long time as two different cultures. And an example of this point of view can be found in the work of Breiman, published 20 years ago. However, in the last 20 years, several changes have occurred, and biostatistics and machine learning blended more and more. Furthermore, I would like to point out the fact that in the last years, several hybrid techniques have been proposed, including aspects of both machine learning techniques and traditional statistical models, especially in the Bayesian framework. But what is one of such blending between such different disciplines? The main reason is the growing use of machine learning for clinical data. Its advantages over the traditional statistical methods, the flexibility and lack of assumptions we have mentioned before, make machine learning particularly suitable to overcome some traditional model challenges, such as the management of nonlinearities, which happen when the relationship between the outcome and the predictor is nonlinear, which is very common within clinical data, or the management of statistical interaction. Another challenge is represented by a few number of observations in front of a huge number of predictors to be tested, which is really common in clinical epidemiology, because we generally work with small, medium-sized data sets in front of a huge number of predictors because of the increasing availability of clinical data used to characterize the patients. We have clinical data, instrumental parameters, but also data from other sources, for example, genomics data or data collected from wearable devices that provide streaming data on the health of the patients, such as about his heart frequency. Traditional models, unfortunately, are well known to have some limitation in managing such issue. There is a lot of literature about this topic, while machine learning provides some solutions that are suitable to overcome limitation of traditional statistical techniques to manage such issue. The growing interest in the use of machine learning models in clinical epidemiology is testified by the number of publications in the field. This is a figure that by a paper published 1 year ago, presenting the number of publications reporting the words machine learning or artificial intelligence and clinical practice, healthcare, and clinical research. As you can see, the number of applications has grown exponentially in the last 10 years. From 2012, especially in the field of healthcare and clinical practice, less in clinical research. Let's see why. What are the main field of application of machine learning in clinical epidemiology? It is important to point out, before talking about the main field of application, that applying machine learning techniques to clinical data may provide new insights that could be useful for clinicians in their everyday clinical practice. And an example is represented by predictive modeling. Predictive modeling is an hot topic, is one of the main fields of application of machine learning techniques, especially in the last 2 years of pandemics, during which a huge number of predictive models was published in order to try to predict COVID-19 diagnosis, COVID-19 treatment, prognosis, and so on. Another promising field of application of machine learning techniques is drug and medical device surveillance. Machine learning techniques are particularly suitable for the analysis of the large data sets used by regulatory purposes, the surveillance of drug adverse events in the post-marketing stage. Disease treatment heterogeneity detection is a promising field of application of machine learning techniques. In 2015, it was published a commentary in Circulation about phenomapping of heart failure with preserved ejection fraction. Phenomapping means to identify a file of patients with the same phenotype, probably differently responding to the same treatment. Machine learning may help in this task because it can help to identify patterns within the data. Another promising field of application is that of clinical research, particularly that of clinical trial design and conduction. But this field of application of machine learning technique is still at the very beginning. We have seen in brief what are the main advantages of machine learning models over most traditional statistical methods that has resulted in a growing interest towards machine learning in clinical epidemiology. But it is important to remember that the application of machine learning to clinical epidemiology presents with challenges. In particular, model rebuilding. This is not the aim of the presentation to talk about technicalities machine learning-based model building. However, it required specific advanced skills and several technical issues to be undertaken. Another challenge is that of causality assessment. The use of machine learning does not any problem of causal reference, because keep in mind that predictors are not causes, while causal inference is a core business of clinical epidemiology. Another problem is represented by model reporting. In the last 2-3 years have been published several systematic reviews that have shown that the reporting of machine learning-based models in the international literature is very poor. So, in the last years, some recommendations have been published for the reporting of machine learning-based models trying to overcome such an issue. Finally, other important challenges in the implementation of machine learning, from my point of view, specific to clinical epidemiology, are the fact that machine learning does not allow to disentangle the effect of each predictor on the outcome of interest, and does not provide the classical measure of association that quantify the relationship between the outcome and the predictor, such as the odds ratio or the hazard ratio, for example. The output of machine learning models is different from the output of a traditional regression model, and for this reason, machine learning outputs are at risk of being perceived as useless by clinicians. And last, another relevant problem of machine learning models is the lack of interpretability. Machine learning is well known to suffer from the black box problem. That is, we can build a machine learning based model that performs very well, but we don't know how it works. And this is a relevant problem, especially in the clinical setting where we work with the patient. As an example, I can build a model based on a machine learning techniques to predict a clinical outcome, such as mortality. The model presents with a good performance, so I propose the model to the clinicians. And I propose to the clinicians to assist his or her everyday clinical practice in order to decide whether or not to admit a patient in the intensive care unit. But how the clinician can trust the model if she doesn't know how the model works? So I think this is a relevant challenge for the application of machine learning to clinical epidemiology. I think it is one of the most important challenges, and I think that several other efforts should be put forward by clinical epidemiologists, data scientists, biostatisticians, in order to try to overcome such limitations of application of machine learning to clinical epidemiology. So, in conclusion, today we are going to discuss about challenges and opportunities about the application of machine learning models to clinical data. I would like to conclude my topic introduction by citing the recommendation of the European Heart Journal. The European Heart Journal recommends to his authors to be as simple as possible, but as sophisticated as needed in the development of their statistical analysis plan. I think that this recommendation is really challenging, and for all of us, clinical epidemiologists, biostatisticians, data scientists, it is really hard to meet such recommendation. So I am wondering if, is machine learning the answer? The question is open. I think that it's time to leave the floor to our today's speakers. I would like to thank you, all of you, for your attention and enjoy the today's presentation. Thank you so much.
Thank you, Giulia, for this very precise and effective introduction. I think it helped to set up the perimeter of the playground for this afternoon. And so I ask Professor Frank Harrell to start his presentation. Frank Harrell is a professor of biostatistics at the Vanderbilt University, the School of Medicine, Nashville, Tennessee. And he's well known for his broad work in predictive model validation, Bayesian modeling, and in general in the broad field of biomedical research, including clinical trials and drug development. So Frank, please.
Thank you, Dario, for the nice introduction. Is my sound okay?
Perfect.
And thank you, Giulia, for setting up the session so beautifully. I'm going to start off by talking about something very controversial. We know that validation of anything we do, whether it's statistical modeling, discovery of genes, or machine learning, validation is all important. There's actually a lot of misunderstandings about what validation means and what kind of validations are necessary. So I want to start off with a very controversial subject, that external validation is overrated. So we know we need validation. It's all important. We need to show that we have predictive accuracy. But there's a lot of uncertainty about what external actually means, and especially if it's geographical or time oriented. So if external means another time or place, it's really not very efficient to validate a model in another time or place, because time or place are expected to be important, and it would be better to model time or place in the original model rather than be surprised by a lack of accuracy in another time or place. So we need to avoid surprises and quit labeling a failure to be accurate in another time or place as a failure to validate. It's really a failure of the strategy that was used for modeling. So we need to learn about geographical and health system differences. We need to learn about time trends and how to get predictions for other times and places not in the data set. If the model is fully pre-specified, external validation validates that model. This is a very subtle point that's lost on most people. If the model is not fully pre-specified, but the model used sort of adaptive analyses such as feature selection, the validation you're doing is not actually a validation of the model, it's a validation of a model. And so when you do feature selection, that process is so unstable that in most cases, all you can get is an example model, not really the set of features. So you're really validating an example model. So in most cases, it's better to validate the entire process that was used to produce the model, using things like bootstrapping, which one of our speakers, Dr. Tibshirani, has really pioneered in this context, along with Brad Efron and others. And while we're validating the process, we can be totally honest about the instability of the process, such as instability of how you select features or models. Here's another controversial idea. I think it's more productive to validate researchers than it is to validate models. And the reason I say that is there's a lot of people wanting to do research, including in large epidemiologic databases and electronic health record, who are really not properly trained, but that doesn't keep them from doing the research. That doesn't keep them from getting it published, because the journals are not qualified to review much of this type of work. So many failures of research findings are very predictable because of bad methodology or inadequate data source. The quality of research and analysis used highly influences the reliability and usefulness of the resulting research. So validating researchers, or at least validating their analyses, is something you can do very, very quickly, more quickly than validating models. So there was a huge scandal at Duke University about 10 years ago related to lung cancer biomarkers, signatures for selection of optimal chemotherapy in lung cancer. And this was called the PODI scandal at Duke. And it was an embarrassing scandal that wasted an amazing amount of resources. And it was really fraud at its base. It could have been averted had the investigators, PODI and Nevin, shared their data and code with an independent group. They refused to do so, which is telling that when people refuse to share those sort of things, there's usually something to hide, and there certainly was here. A great statistician, Lisa McShane, when she got access to the data, which is the funding agency, National Cancer Institute, she had leverage to do that. When they got the data, all hell broke loose because she immediately found severe problems. She even obtained much different results when she ran the code of the investigators using the data of the investigators and ran it twice in one day and got different biomarker signatures from the same code and same data because there was a randomization element in the code, and they were essentially randomizing noise. So an independent research team can check reproducibility and specificity of a statistical analysis plan and can conduct their own analyses to check robustness of the results. Now, Bayesian modeling can come into play and can bring some advantages. So one of the things we use, including methods that Rob Tibshirani has invented, such as the lasso, is penalized maximum likelihood estimation. That allows you to avoid some overfitting, take the data with a grain of salt. So we're discounting the data. And if you're in the frequentist domain, Rob has created a lot of post-selection inference tools that are not used widely enough, but they are perhaps a little complex in some situations. And in general, the frequentist paradigm of standard statistics lacks inferential methods if you're not using standard estimation techniques. So if you're using penalization or shrinkage, you don't really get the same inferential tools. If you're in a Bayesian situation and you use prior distributions that accomplish shrinkage, this leads to plain old posterior distributions that are interpreted exactly the same as if you were not doing any shrinkage. So it doesn't make anything more complex. So you can use sparsity priors, and one popular one is the horseshoe prior. You choose the prior to match the biologic knowledge. So if you're doing a genetic analysis, you would use a prior that matches what's known about the population of the effects of genetic variants over your 5 million variants that you're looking at. And also you need to match the performance goals instead of using convenient shrinkage methods or convenient penalization or choosing something because it runs really fast on the computer. We can choose prior information that matches the problem. And also, if you're in a Bayesian mode, you can do something that's fairly hard to do in the frequentist world, which is to handle ordinal predictors. And you can also have degrees of freedom for nonlinear effects that are data determined and still preserve Bayesian operating characteristics. So Bayesian modeling has a lot of advantages, and it also is a much more elegant way to handle missing data. So instead of having a two-phase system where you do multiple imputation and then you might do 20 separate analyses based on 20 completed data sets from multiple imputation, we can do, and then feeding and then averaging those 20 analyses to get one set of regression coefficients. With Bayesian modeling, you can model the missing data simultaneously with the outcomes. Now, this is a pretty controversial idea, but Bayesian models don't have the same idea of validation because they don't have the same idea of overfitting. So the main disagreements that happen with Bayesian results is if the analyst used something like a flat prior and the reader wanted a shrinkage prior. So to the reader, the effects in the model will be overfitted or overstated, even though they would not be seen that way by the analyst who fitted the model. So when there's a mismatch in the priors, that's when somebody will see it as overfitting. But if the priors are not mismatched, or the reader is using a less skeptical prior than the analyst, then the type of validation that's needed is not the same as the frequentist world. You have all the usual advantages of forward probabilities instead of backward probabilities when you go to Bayes. For example, you could compute the probability that a dose response effect is monotonic. You can compute the probability that a blood pressure reduction is greater than 5 millimeters of mercury. So you have these abilities to do direct probability calculation. Now we get into variable selection, and I call it a mirage because it's kind of a false hope. Unless you have a really clean data set with maybe 1 million patients, and you don't have collinearity. So if those three things held, you can do model selection in a more reliable way, but we don't have those things holding in our world of clinical epidemiology and biomedical research and most areas in general. So the idea of variable selection is you're seeking parsimony, and there's a contest between parsimony and predictive discrimination. Typically, parsimony is the enemy of predictive discrimination. So discrimination will go down if you try to be more parsimonious. Feature selection requires spending information in the data for making binary decisions, and that information can be better used for estimation. The probability of selecting the so-called right variables is 0. It's just something the data are not capable of telling you enough about. Researchers often worry about false discovery rates, and it's very telling how little they care about false negative rates, which means when they make a discovery, there's a lot of non-discoveries that they're missing that really might be important effects, including important genetic variants. The fraction of important features not selected is much greater than 0, and the fraction of unimportant features that are selected is much greater than 0. So the reliability of variable selection is something that's much more problematic than most people will admit. And there's a way to study this and to learn how hard it is to determine how important variables are in a model, whether it's from machine learning or from statistical models. And that is bootstrapping confidence intervals for the variable importance. And if you want to just rank the importance of variables, we're just using a standard bootstrap to get a confidence interval for the rank of the importance of variables. So selecting variables is based on knowing something about how important the variables are. If you cannot learn how important the variables are from your data, you cannot rank the variables, you cannot select variables reliably. So here's an example where you have 300 observations and there's 12 candidate predictors. And the coefficient of the ith predictor is I. And so the beta coefficient for X1 is 1, and the beta coefficient for X12 is 12. So these variables are increasing in importance as you go from X1 to X12. There's a residual standard deviation of 9, and this particular problem is a high signal to noise ratio problem, so it should be easy to select variables. We're going to rank the variables according to their partial chi-square, which would be the same as ranking them on their partial r-squared. And this is the result of the Bayesian confidence intervals. So you can see the variable that was supposed to be the weakest, X1, it came out in the sample to be the second weakest. X3 came out to be the weakest. The variable that's supposed to be the strongest is X12, but it came out the 9th weakest. The variable that came out the strongest is X9, and it was supposed to be the 9th weakest. So it's supposed to be 4th from the top in terms of importance. But look at the confidence intervals. These expose the difficulty of the task. The variable that you think is the most important, which is X9, the data are consistent with it being the 8th weakest variable all the way to the strongest variable. So at least the confidence interval includes the true values in these cases. But to say that the variable that's strongest, first of all, you got the wrong variable. X9 is not the strongest variable, but in the sample, it happened to appear to be the strongest. But to say that the strongest variable, the data are compatible with it being a rank of 8 to 12 is exposing how difficult this problem is of selecting variables. And if you had a lot of collinearities, or if you had many more variables, this these confidence intervals get much wider. So we had a biomarker study where we had 220 biomarkers, and the winning biomarker, all we could say is that the data tell us the winning biomarker out of 220 was not in the bottom half. We just know it's in the top half. We have no idea if it's the winner. And then we can look at reliability feature selection in a different way. This is using the lasso as a feature method. We have 500 observations and 500 candidate variables. So you know danger is about to happen. Anytime this is as big as that, the data really don't have enough information to do what people really want to do. If the outcome variable is binary with an incidence of 0.5 and all the X's are binary with an incidence of 0.1, and we do 2,000 repeats of our experiment and use cross-validation on the deviance to select the penalty parameter in the lasso, and if we sample the betas, the true betas from a Laplace distribution, this is like e to the minus absolute value beta. If you sample from a double exponential or Laplace distribution, that is the prior distribution for the betas that makes the lasso penalty exactly satisfied. So this is giving lasso the optimum performance when the true betas have this sharp peak at 0 in terms of their probabilities distributions. We're going to scale the betas so that we have an ROC area or concordance probability C index of 0.8 for the true linear predictor. So we have a moderately strong predictive discrimination. And for each beta, compute the fraction of the 2,000 simulations in which that variable was selected by the lasso. And you see this pretty frightening plot. So the first thing to look is you have variables that are very strong out here. So their true coefficient close to -1 or +1. What is the probability that lasso selected such variables? So the proportion of the 2,000 simulations for which those variables are selected, it's averaging around 0.5. So where you want to have a probability above 0.95, there is no beta big enough in this simulation such that you had a 0.95 probability of selecting that variable. So we see a lot of 0.5 probabilities, some lower than that. So that means our false negative rate is going to be very high. Our power is low. And these are just not high enough. And likewise, the opposite. What about the betas that were exactly 0 or very close to 0? So that's in here. If you look at the probabilities of selecting, you want those to be close to 0. You'd be kind of happy if they're less than 0.02 or 0.05. But we're seeing an average probability of selection of, if you average all these points vertically, you might get a 0.1 or 0.15. So we have a high chance of selecting variables that are truly worthless. And we don't have a high enough chance of selecting variables that are truly strong. So what you want is a sawtooth pattern where you have a flat line close to 1, and then you have some lowering, and then you want a flat line close to 0 over here. We don't have that. So our probability of coming up with the right set of variables is 0. And now some people just want to use the lasso for prediction. That's a different matter. But a lot of investigators are using the lasso to find out which variables seem to be associated with the outcome, and they're making statements about the identification of such features. And it's that identification of individual features that is the most problematic. So what's the most stable variable selection method? Rob Tibshirani's done some new work that I have not had time to read, and which is to my detriment. But one of the most exciting methods that I've seen is by Pieronen and Vittori, called projection predictive variable selection. This is a Bayesian method where you get a full model that is fitted as smartly as you possibly can, that doesn't try to be parsimonious. And then they have a way of projecting that model onto simpler models. And there is a detailed case study here that goes through this method, and it's really a beautiful example, I think. Now, machine learning, Giulia really discussed this in a beautiful way, and I'll just add a couple of things. What really distinguishes statistical models from machine learning, if you had to pick one thing, is statistical models favor additivity. So we didn't have models where we sort of have an age effect and a sex effect and a blood pressure effect, and those effects kind of add up. And we don't generally put a lot of interactions in our model. So if your method is favoring additivity, it's probably a statistical model. And if it doesn't favor additivity, such as giving interactions equal chance as main effects, then it's probably a machine learning method, such as recursive partitioning, random forest, neural networks, and so on. Statistical models are useful, especially when uncertainty is high or you need to do inference. And then there's kind of a public relations problem with machine learning. Neural networks came out probably in the 1970s. They were touted as having tremendous predictive ability. That did not work out. 10 years later, they came out again with changes and were supposed to have great predictive ability. That didn't work out. And then 10 or 20 years later, it was time to revive neural networks again. And the group got together and said, if we call it neural networks, people are going to remember our past overhype and overstatement. And it's not going to really have good PR. So let's call it deep learning. So deep learning is exactly the same as a neural network with a new name, just for political reasons. Although there's been a lot of enhancements with a lot of better fine tuning and ability to go deeper with more complexity, but it's basically still neural networks under a new name. I have another talk that really goes into detail about what is going wrong with machine learning and some classic fiascos in the medical literature based on machine learning. There's been some mistakes that are so great, it's almost comical. Now, it's very important to choose predictive measures, and there's a lot of misunderstanding about this in clinical epidemiology, tremendous understanding. So we need first of all, this is a gold standard sort of thing you need, is a smooth, flexible calibration curve. You need to show that you have absolute predictive accuracy. And you can't use any binning to get a calibration curve. It has to be a continuous process. The gold standard measure in the frequentist world is the log likelihood. And in the Bayesian world, it's the log likelihood versus the log prior, plus the log prior, which is just your log posterior distribution. And then another great way to think about predictive accuracy measure is explained outcome heterogeneity. And one of the simplest of all ideas, due to a variety of researchers, including Kent and O'Quigley, is just compute the variance of the predicted value. So if you don't have overfitting, the variance of the predicted value really tells you the clinical utility, and you can graph that in various ways. But how different are the predictions? Do you really predict some low-risk patients and some high-risk patients? And I really like the relative explained variation, which you can do it on this scale, the ratio of variances from a subset model to the full model, or you can do it in the relative R² scale or the relative -2 log likelihood scale. So I have this blog article that shows examples of all of these measures. These to me are really gold standard, high information, high sensitivity, accuracy measures, and they're very simple. You don't need anything like IDI, NDI, or any of those other measures. Now, the scary thing is the majority of machine learning papers do not demonstrate adequate understanding of predictive accuracy. And there was a survey not long ago of machine learning applications in medicine, and the survey found that fewer than 1 out of 10 papers even included a calibration curve. I mean, this is about as shocking as you can get for incompetence in demonstrating accuracy of a model and validation of a model. So there's a lot of people doing machine learning that are making the mistakes that we made 40 years ago in statistical models. And I really hate to be living long enough to witness the mistakes that I made 40 years ago being made new. We should be making new mistakes, not making old mistakes. One of the problems is you see a lot of machine learning setting it up as a classification problem instead of a prediction problem. And they're using proportion classified correctly, sensitivity, specificity, precision, and recall as accuracy measures. These are all discontinuous accuracy scores. And so if you optimize them, you'll result in an incorrect or bogus model. These are not valid things to look at if you're not in a pure classification mode. And this also means that ROC curves are highly problematic because every point on an ROC curve, the coordinates are sensitivity and 1 minus specificity, which are discontinuous scoring rules. And importantly, they involve transposed conditionals. So transposed conditional is when you condition on the wrong thing to predict the wrong thing. So you're conditional on the outcome to predict the input. We're going to condition on whether you had a stroke or not to predict your blood pressure. That really makes no sense in clinical medicine or clinical epidemiology, but it's done all the time. And that also invites you to dichotomize the predictors. It's not insightful, and it has a high ink to information ratio. So I've probably seen 10,000 ROC curves in the last 15 years. I have yet to see a single one that provided any insight. Now, predictive measures, if you do things correctly, in my view, that will lead to optimum decision making, and this is all important in medicine. So the optimum base decision might be specified as one that maximizes the expected utility. The expected utility uses the posterior distribution of the outcome probability for a patient combined with the consequences of the possible wrong decisions. And so it really needs to be in a forward mode to use like predicted mortality and things like that. And things like sensitivity and other measures that have transposed conditionals, ROC area, C index. If you look at the expected utility to judge a decision, these measures that condition on the future to predict the past, in other words, they have transposed conditionals, they play no role whatsoever in decision making. So this blog article I mentioned before, looking at measures of explained variation, and we have various ways to graph these things, and I'm just going to show you some examples. And so this is a very simple example where we have age and sex as our basic model, and then we have a proposed new marker, which is total cholesterol. So we're going to use sex by a spline function of age to use our logistic model without assuming linearity for age. We're going to allow for interaction. Our new marker is total cholesterol, which is going to be modeled non-linearly, and it can interact with age, which has been found to be important. And so the simplest way to show predictive discrimination is to show that without the new marker, you have this distribution of predicted probability of disease. So this is the predicted probability of coronary artery disease. This is a back-to-back histogram. And so this is your distribution of risk, which we can call the pretest model, and the variance of the predicted risk is 0.047. If you add cholesterol to the model, by these measures, cholesterol has 17% of the information of the three variables combined. So we have a fraction of the information at 0.17. The post-test variance of the risk is 0.057. You see, we have a little bit more people here in the very high risk, but what has changed the most is we have more people that are assigned very low risk. So these are mainly younger people with low cholesterol are going to have low risk of coronary artery disease. So we've added to this bump here and redistributed our risk. So this is a very, very clinical way to look at added value, way more useful than an ROC curve. Another way to do it is to show your pretest probability. So you're ignoring cholesterol. You've just got age and sex, and you've got your post-test probability, and these are for individual patients. So each point is a patient. How did the post-test probability change once you know their cholesterol? And then we summarize that with quantile regression. This is the 10th percentile of the post-test probability. This is the 90th percentile of the post-test probability. So you can see that knowing cholesterol is going to make the biggest effect when the pretest probability is lower. You're going to move the post-test probability more. If you already are pretty sure the person has coronary disease, you're not going to move the post-test probability very much. And then you can do this in a more insightful way if you wanted to find out what types of patients the predictive model has more utility for. So if you plot the difference in the post and pretest probability, so this is the model without cholesterol, and the post is the model with cholesterol. These are nonlinear effects and interactions. We can see that the difference in pre and post is very age dependent. So as I said before, once somebody's above 60 years old, knowing the cholesterol doesn't help. If somebody's less than 60, the younger they are, the more it helps you to know their total cholesterol. So if you compare this with an ROC curve that hides age and compare it with all the other things you're used to seeing in the literature, all of those things I would wager with you are hiding this age dependency on the utility of the new information, which is here cholesterol. So I'm going to stop there. Maybe we have time for a couple of questions, and I really appreciate your attention. I hope I have irritated you and by just the right amount.
Thanks, Frank, and congratulations for your presentation, very insightful. And we have two kind of a burning questions. One is from Tibshirani, and if he is in already in the chat, he can make it directly. Rob?
Oh, I mean, I can make it by voice?
Yeah.
Okay. Hi, Frank. How you doing?
Hey, Rob, how are you?
I'm tired. It's very early.
Oh, sorry. Sorry.
Very interesting talk, although I only caught about the second half of it. But the lasso example, I was just wondering what the correlation was between the variables that in the one where it did so badly in recovery.
This is where there are no collinearities whatsoever.
So the data was generated with the variables are independent.
They're all independent.
Okay, interesting.
So it's the easiest case.
Thanks. Okay.
Thanks, Robert. And the other question was is also coming from a panelist from the next future days, is Laurie Piniant. I don't know, Laurie, if you are online, if you want to make it by yourself, or if not, okay, Laurie, please.
Hi, Frank. Thanks for the very nice presentation.
Thanks, Laurie.
I was wondering, I of course agree with you that we should look at calibration more often, and specifically because we often have a risk threshold, which we use to make clinical decisions. Now, we also know that this risk threshold is linked to the harms of false positives and false negatives. So that makes me wonder whether classification measures are still as problematic, or the focus on classification methods is still as problematic when those classification measures would take into account the harms of false positives and false negatives. So I would like to hear your thoughts on that.
Yeah, that's something that is, there's a lot of work being done with decision curve analysis that I think is a good way to look at that. And there's a blog on my blog, fharrell.com. There is a blog article about that, and another one is about to come out. And but there's other ways to look at it, which is what we find is if you have a risk threshold, for you to analyze that in your analysis, there has to be clinical agreement about the risk threshold. So you can't just pick a risk threshold saying one clinician uses that. It has to be unanimous among clinicians, or it's not really correct to use that in your analysis. So since we can't get unanimity in the risk threshold selection, I think the risk thresholds are a little bit less useful. And I tend to make predictions that people can apply in the field and apply their own thresholds without them telling me what their thresholds are.
Thanks. Yes, I certainly agree with that. Thanks for clarifying your position.
It's a good question.
So thanks, Frank and Lori, for the interaction. And I will move to the next speaker. I have seen in the Q&A that there are some questions, broader questions. We may discuss them later in the floor discussion. So the next speaker is Ron Summers. He will provide us an overview of the use opportunities for artificial intelligence in radiology. Ron Summers is a senior investigator and radiologist in the Radiology and Imaging Science Department in the National Institute of Health in Bethesda, in Maryland. And he is, of course, dedicated to patient care, but also he directs a lab where machine learning is used to improve diagnosis via radiology. He is a very productive and very well-known author of more than 500 journal papers, and also inventor of holding more than 17 patents. So Ron, whenever you are ready, if you want to start to talk.
Great, thanks, Dario. Can you see my presentation?
Perfect.
Wonderful. Well, it's a great pleasure to be here today. I'd like to thank Dario for the invitation. And I think it's also a real pleasure to be on the program with Professors Harrell and Tibshirani. So I'm a radiologist and I run an AI lab at the National Institute of Health Clinical Center. These are my disclosures, by the way. This is the NIH Clinical Center, which is the world's largest hospital devoted solely to clinical research. And I'm going to be focusing on epidemiology using AI for abdominal radiology, but to put this into context, there are opportunities for epidemiologic clinical studies throughout radiology, and these are becoming more popular as machine learning is able to help create the data needed for such studies. So for those of you who may not be familiar with abdominal radiology, it is a clinically highly important subspecialty of diagnostic radiology as a whole. It has its own society, the Society of Abdominal Radiology. The abdominal radiologist is responsible for identifying disease and characterizing it in numerous organs, and there are numerous lesion types in the abdomen that make it a very challenging field. Now, in regard to epidemiologic studies in radiology, these have traditionally been limited to screening applications, most commonly for breast cancer imaging and mostly mammography, as in this mammogram example on the right. So probably there have been hundreds, if not thousands, of epidemiologic studies of mammography and its ability to detect cancer and issues such as unnecessary biopsies and anxiety caused by false positive results and so forth. And much of the remainder of radiology has been somewhat free of epidemiologic investigations. And there are a number of reasons for this. That has to do with lack of reporting standards and diagnostic guidelines. One of the reasons mammography has been a popular topic is because the reporting of mammography studies was standardized a while back using the BI-RAD system. And these reporting standards and diagnostic guidelines have made it easier to conduct these studies. There's been insufficient computing power. GPUs are now widely prevalent, so that's less of an issue. There were inadequate algorithms, as we heard from Giulia and Frank. These algorithms have been improving greatly, particularly since about 2012. A big issue has been the reliance on manual measurements in radiology. Manual measurements are extremely time-consuming in radiology, and they also require expert knowledge in order to make the measurements correctly. And for that reason, it's hard to do very large studies because manual measurements on large studies are just intractable. They're expensive and inefficient. There's been a limited availability of public data sets. This has changed quite a bit, particularly over the last 5 years, and the number of large data sets are increasing annually. So all these limitations have changed, and we're now in an era where epidemiologic studies using radiology data are becoming much more tractable and much more commonplace in the literature. As a general point, following along with what Giulia showed in her introduction, the use of deep learning or machine learning or AI in radiology has really been taking off. You can see from this graph that there's been, particularly over the last few years, an exponential increase in the number of papers being published in the field of radiology. But for abdominal radiology, you can see the number is much smaller, and that makes it a great area to focus on for those interested in a less well-studied but a critically important topic. Now, my lab has been working on image processing of radiology images for many years, and this mosaic of images shows some of the different clinical problems that we've been investigating, including precancerous polyp detection in the large intestine, spine localization and abnormality classification, osteoporosis detection in the spine, which is particularly useful in the elderly, early metastasis detection, such as this epidural mass in the spinal canal, which is one of the frequent early places where cancer can spread and can lead to symptoms such as paralysis and loss of bowel and bladder function. Small bowel imaging is a relatively less well-studied area. Liver, spleen, kidney, aorta, and adrenal imaging are starting to become of great interest, particularly in the machine learning community. This is a kidney with multiple tumors detected by an automated segmentation algorithm. And this is an example of a cancer patient with an abnormal lymph node shown in purple here, detected by an AI system. So lots of different opportunities for AI. But in this presentation, I'm going to focus on one particular application where my group has focused, and that has led to a number of epidemiologic studies that we published over the last few years, and that's on body composition. So in the clinic, we know that certain elements of body composition are predictive of things like cancer and heart disease and diabetes. And clinicians are very used to making measurements like body weight, waist circumference, body mass index, and lean muscle mass. These are very easy to measure. They're non-invasive metrics, and they have been used as surrogate biomarkers for health and risk of disease. And there's been an expanding role of these body composition metrics in clinical research and practice, particularly in the US, where we have an obesity epidemic. The projects that I've been doing on large-scale body composition analysis have been with my colleague Perry Pickhardt, shown here. He works at the University of Wisconsin-Madison, where he's the head of abdominal radiology. This project is in year 10 and has led to 22 clinical papers that have combined both traditional image processing and deep learning. The particular studies that I'm going to show you used 5 AI tools, and the number of these tools are increasing, and we're continuously improving them. The 5 tools are shown here. The first is an abdominal atherosclerosis tool that identifies calcified plaque in the abdominal aorta. The second is one that analyzes the liver to assess for fatty infiltration. Fatty infiltration leads to the liver looking darker on a CT scan and can lead to cirrhosis and hepatocellular cancer, particularly if it's a long-standing problem in a particular patient. This image through the abdomen at the level of the kidneys shows analysis of the muscle and I'll explain how a number of studies have shown that muscle quality is a good predictor for important outcomes like overall survival and cardiovascular morbidity. This is an image showing bone mineral densitometry on routine CT, again relating to osteoporosis. And this example on the lower right shows visceral and subcutaneous fat analysis. Visceral fat is particularly important in diseases like diabetes and metabolic syndrome. So now I'll talk about the bone mineral densitometry tool. Now, traditionally, the way bone mineral densitometry is measured is using a technique called a DEXA scan, or dual energy X-ray absorptiometry machine, which produces an assessment using a low radiation dose. But bone mineral densitometry can also be done on routine CT scans of the abdomen. And this is a close-up of one of the bones in the lumbar spine, where the measurements of bone mineral densitometry are being made on what we call an opportunistic basis. And this graph on the left shows a comparison of manual and automated measurements. You can see the automated tends to slightly overestimate the bone mineral density, but that's probably because of the specific location in which the measurements were made. This example shows a study we did back in 2011, where we looked at CT scans done within 2 CT scans done supine and prone on patients with, these were done for colorectal cancer screening, where the patient underwent 2 scans within about 10 minutes of one another. And we were able to show reproducibility in this Bland-Altman plot with a reasonably low difference between the supine and prone assessments. This particular method used some traditional image processing techniques rather than deep learning, including a watershed algorithm, spinal canal segmentation, and a directed acyclic graph method, as well as a vertebral level template combined with an active contour method. So you can see that many of these automations can be done using traditional image processing techniques and can be quite accurate. The way this method identified the individual bones was to take a curved planar reformation of the CT scan and then identify the intervertebral discs, that those separate the spine into the individual bones, and that enables 3D reconstructions of the spine, such as this image on the right. The second tool that we developed looked at the visceral fat, as I mentioned earlier. This is an example of a patient in 2007 who had a large amount of visceral fat, shown in blue here, and in 2017 had considerably less visceral fat. What we found in a population-based study of several hundred participants was that the annual rate of change in the visceral fat tended to be higher in the men than in the women, compared to changes in subcutaneous fat, which had an opposite trend. And these, of course, are well-known phenomena, but it's now possible to do this in large patient populations and to identify more interesting and potentially less obvious but pertinent factors. The adipose assessment is something that we've been doing for over a decade. It's extremely useful in different patient populations. For example, patients on different drug therapies have sometimes different amounts of visceral fat, and we've also found some correlations with colonic polyp formation in visceral fat, which I'll mention a little later. The adiposity assessment, again, is a traditional image processing algorithm that uses anisotropic diffusion for smoothing, fuzzy C-means to put pixels into a body and background class, and then a repeat fuzzy C-means to classify the adipose and non-adipose tissue. And then an active contour model separates the subcutaneous and visceral fats components by identifying the outer boundary of the internal body cavity. So these are Bland-Altman plots showing the difference between human observers and the computer method, and the human observers showing similar differences between the two observers, whether they're the computers or the computer or the humans. We had a difference of about 1 or 2% in the measurement. So a highly accurate method for assessing adiposity. The third tool that we developed was one on detecting abdominal aortic plaque. This method used region proposals to identify the location of the abdominal aorta. The plaque is this white material here. The spine is here, and parts of the liver and spleen are visible in this particular image. And this is what the image looks like originally. This is a preliminary segmentation of the plaque and then a final segmentation of the plaque. And many of you may be aware that a plaque analysis is usually done on the coronary arteries in the heart. We've been looking at the utility of extending this to the abdomen because not everybody gets a chest CT or a cardiac CT, and having the ability to measure this plaque in other body parts may prove clinically useful, as we found in some studies. So here's a look at a group of about, I think it was about 9,000 patients looking at atherosclerotic plaque over the lifespan. You can see that using these 3 different types of plaque analysis, which are commonly used in the heart, you can see around the 6th decade of life, the amount of plaque just starts taking off very rapidly. And as is well known, men tend to have more plaque than women, as shown in this bar chart. This plaque tool is a deep learning based tool. We initially developed a 2D tool that identified the plaque, but also came up with many false positives, which needed to be reduced. The way we did that was by developing pseudo classes, by clustering the CNN features. This enabled us to distinguish the plaque pixels from those relating to other bright spots on the CT scans, like the ribs and non-vascular related calcifications. And then we retrained a convolutional neural network model using the true plaque class and pseudo classes. So here's some plaque in the abdominal aorta. Here's a false positive on a bone spur in the spine. And in these 2 different examples, we can see false positives. This one's on a muscle here, were eliminated with this technique. And you get the final segmentation here, which can then be used to measure the plaque burden in the patients. The 2D method used a mask RCNN with a ResNet 50 backbone. It was initially trained on low-dose non-contrast CT scans of 112 patients in which all the plaques had been manually measured, and it had 85% sensitivity at 10 false positive plaques per patient. We subsequently improved the method using a 3D deep learning algorithm and extended it so that the method could work not just on non-contrast CTs, but also contrast-enhanced CTs. Here are examples of plaque on a sagittal image showing the spine, the aorta, and you can see the thin plaque. Here's the segmentation of the plaques, and the images on the bottom show non-contrast and contrast-enhanced scans of the same patient in which the plaque is accurately segmented. This improved 3D method used a modified 3D UNet with a dice loss. It was trained first on the same 112 non-contrast low-dose CT scans. We added 23 CT urography scans to distinguish a common false positive of contrast material in the ureters. Those are the tubes that go from the kidneys down to the bladder. This method was tested on 922 CT colonography or virtual colonoscopy scans and 1,207 paired non-contrast and contrast-enhanced CT scans from renal donors. These are normal people who are donating their kidney and had a CT scan for evaluation of their kidney prior to the transplant. And this method had high correlation and agreement with the manual measurements, showing that it worked well on both the non-contrast and the contrast-enhanced scans. The fourth tool we developed was an abdominal skeletal muscle tool. The skeletal muscle is this tissue here surrounding the abdomen on this abdominal CT slice through the mid-abdomen. There's also muscle adjacent to the spine here. This is called the paraspinous musculature. In red on the right, you can see the automated segmentation of this muscle. The image on the bottom shows a patient with poor quality muscle. You can see that the muscle thickness is less compared to in this patient. You can also see that there's fat. That's this relatively dark area within the muscle and the paraspinous muscles, indicating low quality muscle. And the algorithm works well on detecting that muscle as well. In a similar study over the lifespan, we found that the mean cross-sectional area of the muscle tended to be relatively flat until later in life when it started decreasing, but you can see a greater rate of decrease in the muscle density, which is proportional to the amount of fat appearing in the muscle. And studies are starting to come out showing that this muscle quality, known as myosteatosis in the literature, turns out to be a very important risk factor that has been poorly understood, again, showing the kind of benefit that large-scale studies with machine learning can add to patient care. The graphs on the right show that the phenomena of decrease in quality and cross-sectional area were similar in men and women, with men tending to have a higher baseline muscle and quality. So this particular method, dubbed sarcopenia assessment, sarcopenia being the term, medical term describing decreased muscle mass, can identify individual muscles and muscle groups as shown in these examples. It can also identify individual muscles or clusters of muscles as shown in these images. The sarcopenia tool is a deep learning method. It used a 2D UNet. Was trained on 102 CT scans that were manually segmented with an equal male to female ratio. These were consecutive cases and they were using contrast enhanced CT. These plots show the dice similarity coefficients for the accuracy of the muscle segmentations in different muscle groups, including the paraspinous, psoas, and total musculature. You can see the dice coefficients generally are above 90%, which is considered a clinically useful accuracy score, although we did notice that the dice coefficients had a trend to be higher in patients with greater amounts of muscle. The fifth and final tool that I'll mention is one measuring CT volume and CT attenuation. Traditionally, radiologists measure the size of the liver by measuring the maximum span of the liver from the bottom to the top. The lungs are up here. This is the bottom of the heart. This is a coronal image of the body from a CT scan. I think you can appreciate, because of the complexity of the shape of the liver, this single linear top to bottom measurement is not a very good one, and yet that's what radiologists have used for decades since the development of CT, and even prior to that when ultrasound was used. But today we can now do volumetric assessments much more easily with deep learning, and it's believed that those are more useful indicators. The other thing that we can measure besides volume is the average density of the liver. This is a patient who at initial CT had a density of 42 Hounsfield units, Hounsfield units being the measure of pixel intensity in a CT scan. 5 years later, that dropped to 30 as fat started infiltrating this patient's liver. And 10 years later, this patient had a lot of fat in the liver. You can see it's darker here, and the measurement was 2 Hounsfield units. The plot on the left again shows a comparison of manual and automated measurement of liver attenuation in, I think this is about 5,000 CT scans. That's a lot of manual measurements. And you can see the automated algorithm did quite well. And in this confusion matrix, there was some off diagonal errors, but they tended not to be too bad. The nice thing about some of the deep learning techniques is that using techniques like generative adversarial networks, we can do domain adaptation and extend techniques developed for contrast enhanced CTs to non-contrast CTs and vice versa. This is very important because CT, as well as other types of radiology images like MRI and PET scans, can be done in many different ways, and we don't want to have to be in the business of manually creating ground truth data sets for every potential variation of scanning. This liver segmenter worked using a 3D UNet with a cycle GAN to convert the contrast enhanced scans to synthetic non-contrast CTs and was trained using 231 contrast enhanced CTs from a public data set called the Liver Data Decathlon. And this technique worked well in the in-distribution contrast enhanced scans with Dice coefficients in the mid 90%, and on out of distribution non-contrast CTs, the Dice coefficients were in the high 80%. Okay, so now with all that background, I'd like to tell you a little bit about an epidemiologic study that we did using these 5 different AI tools. This particular study was published in Lancet Digital Health 2 years ago. It used the 5 different tools on a set of CT scans from 9,000 patients who had come to a single medical center for colorectal cancer screening, so they were otherwise an asymptomatic population. We looked at the ability, I'm embarrassed after Frank's talk to show ROC curves, but this is the mainstay of work in radiology on ascertaining the value of these different AI tools. But we looked at the aortic calcification and the muscle density and found that the areas under the ROC curves were higher than those of the traditional Framingham risk score and body mass index, which are 2 non-invasive methods used to predict overall survival. And when we used a multivariate model using the calcification in the aorta, the muscle density, and the liver density, we obtained an even higher area under the curve of 0.811. This example shows the sort of prediction that's possible in individual patients. This was a 57-year-old man who underwent a CT scan that showed a high visceral to subcutaneous fat area ratio, a low liver density indicating fatty infiltration, and a relatively high abdominal aortic plaque score. And 12 years after the original study, his liver had increased in fat, his aorta had increased in atherosclerotic calcification, his visceral fat had increased, and his muscle quality had decreased. And this patient died shortly after this CT scan. Another epidemiologic study we did using this same data set and 5 AI tools was the prediction of future major osteoporotic fractures. This is, again, an anecdotal example of the kind of thing this study was able to do in a large patient population. This was a 59-year-old asymptomatic woman who 12 years prior to developing a hip fracture had a bone mineral densitometry of 135 Hounsfield units, which is in the low normal range. After 4 years, that dropped to 92. Another 5 years later, it dropped to 72, then to 63. Her muscle attenuation was quite low, less than 0, which indicates substantial fatty infiltration. And then shortly after, she developed a fracture. Now, in radiology, I've shown how AI can be used to do these relatively large epidemiologic studies, but as the data set, the radiology data set gets larger and larger, it gets harder and harder to create AI tools that are accurate, particularly in different patient populations. And so we've been exploring different ways to train AI systems using already existing data that can be extracted from the patient's medical record. So this was one of our first attempts. It involved taking 780,000 radiology reports and 216,000 key images. These were images that radiologists during their normal clinical work were reporting as important as showing a site of disease. And we fed both the images and the reports into a convolutional neural network, which was then able to predict from a single input image some labels for that image. So for example, this patient with a kidney cancer, the AI predicted that there was a mass in the kidney. Subsequently, we developed a publicly available data set we call the Deep Lesion data set. This consists of CT scans of over 32,000 lesions from over 4,000 unique patients. These are lesions throughout the body, and the data set includes the location and measurements of the size of the lesions. And we use this data set and annotations to develop a universal lesion detector that can identify lesions throughout the body, including masses in the neck, nodules in the lungs, liver lesions, and lymph nodes and spleen lesions. Another public data set that we developed was one showing abnormal lymph nodes in 176 patients. You can download this data set by searching for the Cancer Imaging Archive CT lymph node, and there's over 1,000 lymph node annotations, candidates, and masks in this data set. And this particular data set is being used to develop AI systems that can detect and size lymph nodes. We've extended that recently to MRI scanning. This is work that just came out at one of the MICI workshops last year, looking at MRI scans, identifying lymph nodes in different parts of the body. And you can foresee applications of this technique for choosing oncologic drug therapies. In clinical practice, radiologists and other physicians need to make measurements of lesions on the scans, like this mass behind the spleen on a CT scan of the upper abdomen. This is an automated measurement using an AI algorithm, and it's comparable to that of 3 different manual assessments from 3 different observers. So again, another way that machine learning can help with standardization, perhaps, in oncology analysis. So in my remaining time, I'll just mention some other techniques that can be used for epidemiologic studies using radiology. This is comprehensive spine oncology analysis, looking for metastases in the bones in the spine. We've been looking at pancreas lesions with this public data set that we made available of 80 CT scans, enabling segmentation of the pancreas, 3D modeling, and identification of cancers in the pancreas, a very problematic disease with poor prognosis in most patients that suffer from it. And we've also looked at phenomena on the scans that make organs like the pancreas easier or harder to segment, in this case, depending on the amount of visceral fat present in the scan. Some other applications include bowel imaging. The small bowel is a relatively difficult area for AI to analyze. We've been improving that iteratively over the years, enabling detection of small bowel tumors and small bowel obstruction potentially. In colonic analysis, colon cancer being the 2nd most common cause of cancer death in America, at least, we've been looking at the impact of visceral fat on polyp formation in the adjacent bowel and looking at some trends in the location of polyps based on the amount of visceral fat adjacent to the bowel under the hypothesis that there might be a a local effect of this metabolically active visceral fat. Another thing that we can do with machine learning is link different types of imaging studies together. This is an example of a technique that can link endoscopic imaging with CT colonic imaging by mapping the center line of the colon on an endoscopic scan to that of the center line of the colon on the CT scan. We can enable correlations of the endoscopic images with the CT scanning, and then precisely locate findings on the endoscopy with those on the CT, enabling some interesting correlations. So I'll just conclude with some challenges and questions. Some of these were alluded to in Giulia's introductory talk. This issue of interpretability and explainability are of great interest and may impact clinical implementation of AI. There's continuing brittleness of AI systems, particularly due to domain shift, some of which can be ameliorated using these generative adversarial networks. And there are also issues of ethics, bias, and trustworthiness of AI, which are of great interest to society at large. There have been many lessons from the COVID era. We need, particularly in the radiology machine learning community, to avoid duplication of effort, and clinically relevant data and endpoints are key. And if you'd like to learn more about these two topics in the COVID patient population, I encourage you to take a look at my editorial in Radiology last year. Some approaches to using less labels to train AI systems and extend the reach of epidemiologic studies using radiology imaging include meta-learning, such as zero and few shot learning, and other types of learning, including transfer, unsupervised, semi-supervised, and federated learning. So to conclude my remarks, the abdomen is a relatively less studied area for AI in radiology and for epidemiologic studies. There are practical clinical benefits for AI in abdominal radiology and many opportunities for clinical epidemiology using radiology. The studies that I've described benefited from the participation of many different investigators and funding sources. If you'd like to learn more, I encourage you to check out my website and our GitHub site, where you can download code and some AI models. And I'm looking forward to your questions and to the panel discussion. Thanks very much.
Thanks, Ron. It's very amazing, the results and that you have shown us. I think I have seen there are some questions in the Q&A, but because I feel they are opening the field to a broader discussion, we move them to the panel discussion. And I know that Giulia is willing to ask you some. Giulia, you can make it direct. You don't need to.
Yeah, there is a question, just a small question, asking for what opportunities are there in radiology for other types of epidemiologic studies?
Right, so I've focused on abdominal imaging. I mentioned at the beginning of my talk that breast imaging is an area that's traditionally been a focus of epidemiologic studies. We're starting to see quite a number of large-scale studies in the brain and chest, for example, for lung cancer and for different types of brain cancer, as well as for neurodegenerative diseases like Alzheimer's. Another area that I've worked in, we presented some work recently at the Radiologic Society of North America meeting just a couple of weeks ago, was looking at diabetes. We performed a large-scale study of diabetics and found some interesting trends in the appearance of the pancreas on CT scan. So I think a lot of these studies are in their infancy, but as the data sets get larger and the number of applications expand, we're going to see quite a few more of these studies.
Thank you so much for your answer.
Thank you, Ron. And if we may move to the last speaker in this afternoon before the panel discussion is Robert Tibshirani, and who is a professor of biomedical science and statistics at Stanford University. And he has been already mentioned for his contributions like the lasso, the bootstrap, and also for his work in significant analysis of microarrays. He has authored several books that we all have studied. And so, Rob, please, whenever you are ready. The audio. Yeah.
So thank you, Dario, and thank you to the committee, the organizers for this invitation. I wish I could come in person. I've never been to Padova. Looks like a beautiful place from the pictures I see on the web. Maybe someday.
You will, for sure.
So let me share my screen. Okay. Can you see that? Let's see. You can see that, I hope?
Yeah, it's perfect.
Okay. Actually, let me see if I can go full screen here. Okay, so I'm going to give a sort of a general talk about data science, statistics, and health with a focus on sparsity. Well, it's going to be general at the beginning, then it'll kind of become more specific with some specific new methods and examples I'll talk about at the end, time permitting. So the outline, I'll talk about some general thoughts about data science and health, some general advice for data scientists. You know, as you get older like me, you tend to want to give advice more often, so that's what I'm doing. Some comments about supervised learning and sparsity. Then I'll give a couple of examples. The first one of GWAS, genome-wide association studies, and some new methods for being able to model those with a lasso extension. And then what I call the elephant in the room, deep learning, which both the previous two speakers have mentioned, Ron especially shows some very nice examples of this application. And then something called LassoNet, which is, as you can guess, is some kind of marriage between deep learning and the lasso, some very recent work. So I'm just going to start to say the last time I heard Frank talk with Stanford 2019, actually in person, and he was, what I say, grumpy, but in a, grumpy in a nice way. He was critical of big data analysis and machine learning, which he was again today. Okay, so, but that first talk, at first, when he first gave that talk, it was like, I was a bit taken aback, like, why is he being so critical? And then I listened more carefully what he said, and I asked for his slides, and it led to a paper. He was complaining about, for example, in that work about cross-validation. He doesn't believe some of the answers people get from cross-validation, and he was right. So it led us to sort of to rethink this cross-validation. And a very recent paper I've completed with Stephen Bates and Trevor Hasty, cross-validation, what does it estimate and how well does it do it? Derives proper standard errors and confidence intervals for the cross-validation error. And that's on arXiv. So I'm not going to talk about this, but I just want to say, when Frank speaks, you should listen. And I listened again today, and I'm going to look forward to seeing his slides because he often makes very insightful comments. They're often critical, but that's good if they lead to improvements in methods. So, okay. So in general, there's a lot of excitement, of course, about data science and health.
Rob, sorry to interrupt you. I don't know if it is meant for purpose, but you are sticking on the first slide.
I'm sticking on the first slide?
Yeah.
Really? Okay, so still first slide now?
Yeah, still.
Second slide? No, okay, so I was already on slide 3. Maybe, I wonder how that could be. I'll try to maybe share. I'll do a new share. Let's see.
Okay, now you moved.
Well, now I moved because I stopped sharing. Let me just.
That's okay.
let me share again. See, maybe I'll share my entire desktop. Maybe that could fix it somehow. So, what slide do you see? Do you see the first slide now?
This is the first one, yes.
Yeah, let's see if you see this. Okay.
Yeah, now it's moving.
Now it's moving. Except I wanted to use. Okay, well, that's okay. I'll figure this out. Okay, now it's moving. So, okay, so you didn't see this slide. That's okay. I read it basically. And then I mentioned some comments about Frank and his, last time I heard him give a talk, which led to this paper about cross-validation, which I just put here for your interest, which is about deriving proper standard errors and confidence intervals. You can find it on arXiv. First author Bates. But I'm not going to talk about this paper in detail in this talk. got lots of other stuff to talk about, but I think it's very useful. And again, I thank Frank for his talk, which led to this paper. Okay, do you see slide 4 now?
Yes. It's working now.
Okay, so I guess I've sort of said, well, I started to talk about this, data, and we saw, for example, in Ron's talk, some of the successes of especially deep learning in radiology and other, as example, and it's also been successful in other areas like other areas of pathology and other diagnostic specialties, especially. And it's now, of course, getting huge amounts of data in many areas of health and medicine, from smartphones to smartwatches, and et cetera, et cetera. So there's a huge amount of data coming in, and some very exciting new tools for trying to use them to answer questions. And for statisticians, of course, you've heard of this. The 2009 Hal Varian at Google said that statisticians have these, the sexiest job for the next 10 years will be statisticians. I guess our time has expired, but maybe we'll get a new quote. Harvard Business Review agreed in 2012. So we need a new quote. I don't know if sexy will be involved, but certainly there's a huge amount of interest in data scientists. Companies of all sizes, not just places like Google and Facebook, but startup companies, right? Or small companies are collecting data at unprecedented rates and trying to use these data to improve their products. Whoop. Okay. So some obstacles to the success. Well, in the US especially, data siloing is a problem. And that's refers to the fact that the US healthcare system, researchers tend not to share data. So, when you're studying like say a certain kind of cancer that's relatively rare, one of the big bottlenecks is you can sometimes not get access to large data sets because everyone has their own data and it's worth money, right? It's proprietary. And we need access to more data in order to try to answer questions well. So that's a problem in the US healthcare system, not so much in, say, Scandinavia. I'm not sure how it is in Italy or in Europe. Canada is better because we have this, I'm Canadian, so I know this. I mean, they have a universal healthcare system and they share data in a much more centralized way. Another fact is that the bar is higher in health in data science, right? There's more at stake than if you're saying a recommendation system for movies, where if you make a mistake, someone has to watch a bad movie. Whereas in healthcare, of course, you make a mistake, the consequences can be much more costly. And the public is less tolerant to data science errors than human errors, right? Analogous to self-driving cars, right? Where self-driving cars, at least at Google, for example, are far safer than humans in terms of the number of accidents per mile. But when you hear about a single accident involving a self-driving car, everyone panics. This is terrible, right? Despite the fact the cars are in general much better than us humans at driving. And another obstacle, which I think has unfortunately come to the fore in the last couple of years with the pandemic, is that building trust in data and science is really much harder than previously thought, right? Probably a lot of you share my feeling that I'm shocked at the lack of knowledge and trust in science that we've seen among large sectors of the population in the last couple of years. So that's an additional challenge. Okay, whoop. Okay, so you see slide 7. Yes? Dario?
Yes, yes.
Yeah, okay, I'll stop asking. I just want to make sure that if it seems to be stuck, let me know in the future. Okay.
I cannot see the number of the slide. So this slide is high-level comments.
It's
7. Okay, yes.
Too small. There it is, but okay.
Okay.
Okay, so high-level comments about data science, statistics, and machine learning. I think the first thing, which I think a lot of you will agree, and Frank certainly hit upon this point as well, that data science and statistics involve much more than running a machine learning algorithm on data, right? Although a lot of people in machine learning, they think that is statistics now, or data science, right? They grab the data, they run a machine learning algorithm, the results look good, and they want to publish it or give it to a collaborator. But of course, that's not realistic, and it's even dangerous, as I think Frank also mentioned this. So, for example, what exactly is the question of interest? How can I collect data or run an experiment to address this question? sometimes I work with a lot of investigators in the Stanford Medical School, and sometimes they'll come to me with a data set they've collected, and they want me to analyze it. I ask them what the question is, and then I realize this data can't answer that question. So I'll send them back and say, look, this isn't realistic. You need to collect, your data's got a problem. It's too small. It's sampled the wrong way. You've not measured the right variables, right? So that's an important skill a statistician, a data scientist has to learn, to figure out just what the question of interest is, and and how to collect data to run an experiment to address the question. Now, having run the experiment, what can you infer from the data? What action can I take as a result? Do I need to worry about bias, confounding, generalizability, concept drift? So this is, again, this is a plug for statisticians, and I'm guessing a lot of you are biostatisticians. These concepts, bias, confounding, these are old concepts that are not going to go away, right? No matter what algorithm someone comes up with, some fancy algorithm for analyzing data, they have to worry about bias, confounding, generalizability, concept drift, and more. So for this reason, the kind of concepts we learn as statisticians and the training is essential for anyone looking at data. Okay. This is a slide that's a colleague gave to me, which I think is kind of funny, but also telling. Who wants to become a data scientist? Oh, everyone wants to. Who wants to learn programming? That's okay. It involves a lot of work. Who wants to learn statistics? And they're all gone. Okay. So, this is, I think, it just makes the point that being a good data scientist means you, of course, need to know software. You have to have the software skills, but you also need to learn statistics. Okay. Okay, some important points for data science applications in health. Models should be as simple as possible. And why is that? Well, they make it easier to understand by you and by your collaborators. We can also more easily anticipate when they're going to break, right? If you fit a neural network to some data and it works well on your current data, and then you or someone else tries to apply it tomorrow to a similar domain, it doesn't work anymore, predicts poorly, say. Figuring out how to, why it broke and how to fix it is quite difficult because it's a complex model. So that's a push for simplicity, both for understanding and also being able to adapt models in the future, which is always necessary. Now, I'm not going to be as critical of neural networks maybe as Frank was, because I'm going to talk about neural networks in a bit. They have their role, and they're really, in some ways, very exciting what they've done. And we saw some examples from Ron. But it depends on your objective, really, right? If you're trying to predict, then neural networks are very useful, and I'll talk about that. But if you're trying to interpret a model, they're not as attractive. It's important to quantify uncertainty, right? Not good enough just to fit a model and say, look how well it predicts. How much certainty do you have in that prediction? And that's come to the fore in, say, in COVID work. We're trying to forecast COVID cases, and I've had personal experience with this. It's very difficult. You can make a point estimate, but if the confidence intervals or prediction are so wide as not to be useful, then the forecast is not useful. And unfortunately, that is often the case when you try to, we found, when you try to forecast COVID, because it's extremely, the data is very noisy. The whole process, of course, is extremely messy. And so you need to quantify your uncertainty, which is often large. So large, again, that the forecast might not be useful. Fairness in algorithms is important. Here's other examples of a key unsolved problem. In classification with high dimensional features, how can you arrange for a classifier to sometimes abstain? Right? Sometimes, and I have experience with this, where I work in people oncology, where we're making predictions from large sets of proteins, predictions of whether someone has cancer or not. But sometimes you have a training set, you build your model, and then a new patient comes in, and their feature vector is so different than your training set that the model has to extrapolate. So we want to arrange for a classifier to say, sometimes I abstain. I can't make the prediction. The question is how to do that in sort of a systematic way that you can quantify the resulting properties of your procedure. And I've made some progress with that with some student. We published one paper, but it's not solved yet. Another example that's getting a lot of attention is heterogeneous treatment effects for personalized medicine, right? Given a patient, who are trying to decide between two drugs, which drug to the patient, which one is more likely to give you a treatment response? Well, this is a hard problem because you never get to see the outcome under both of the drugs. So, example of a simple unsolved problem, how do you do cross-validation for such a model? Because you don't get to see the why for everybody. You get to see the response with one or the other treatment, whichever one was given. So this is a fundamentally harder problem than the usual sort of supervised learning, but extremely important. Getting a lot of interest now in not just medicine, but also economics and business and lots of areas. Details matter. So this is this tends to be kind of my experience also working with people at Stanford University and also in companies. Have you tried method X? I tried that last year, it didn't work very well. I get this very often from collaborators. What do you mean? What exactly did you try? How did you measure performance? I can't remember. The details matter, right? And the details matter more and more now as the methods become more complicated. What algorithm did you use? How did you deal with outliers? How did you estimate the tuning parameters? How did you do the validation? How did you measure the performance? Again, classification, calibration. Details really matter, and more than ever. Some general tips then: try a number of methods and use internal cross-validation to tune and compare models. So this is one thing. We have a luxury as data scientists that we can do experiments, try different methods, and the computer is our lab. It's essentially free and it's very fast, right? Imagine people that are in the wet labs and they're trying to come up with a new method for doing something. They can spend months and months in the lab. They have to, right? And maybe after 6 months, the method they tried doesn't work, and they're back to square one. We have a luxury of our lab as the computer. So one should be systematic when you run different methods. Try lots of things. Be systematic. Use internal validation or cross-validation to compare methods on the same training and test sets. Keep carefully documented scripts where practical, and data archives. The point is your work should be reproducible by you and others even years from now. Right? I have, sometimes I look back, if I've done analysis and I haven't documented it properly, best data comes back a year later. Oh, Rob, remember that work we did a year ago? I got some more data, and can we do this? I look back, I can't remember what I did. I have, because I didn't document it properly. So it's really important, both for you and your collaborators or other people, other researchers, to document carefully what you do. So you'll remember, and so the details are there. Other people can try to reproduce it, can critique it, can tweak it. Very important. Whoops. In praise of simplicity. So this is my last philosophy slide, and then I'll drill down and be a little more specific. But again, I want to push on this simplicity point. So many times I've been asked to review a data analysis by a postdoc in biology or company employee. Almost every time they're unnecessarily complicated, right? The postdoc will say, well, thank you for talking to me, Dr. Tibshirani. Here's my data. And then I'll have 30 minutes of, I applied this algorithm from this package, and then I did this, and then I drew this picture. And after 30 minutes, they ask, so what do you think? And of course, my answer, I have no idea what to think because it's so complicated. It's not calibrated. You made 10 arbitrary choices along the way, and I can't tell how much how important each step was. I can't tell the other things they tried that didn't work. And it's very complicated. Well, I was thinking about that. Why do we sort of see so many complicated analysis? And I think it's kind of a social point. And I'm guilty of the same thing. We have advanced degrees, a lot of us, right? Masters, PhDs. And we'd like to justify our education. We can't just fit a regression or do a plot. That's too easy, right? So we tend to do things complicated sort of because we've learned complicated things and we think we're advanced scientists in the field. We need to do things complicated. Well, actually, if you can come up with something simple that works and is explainable, that's a much better achievement than doing something that's complicated and appears to work. So don't be ashamed of simplicity, be proud of it. And then there's the hit everything with deep learning problem, okay, which I'll address a little more later, right? Now deep learning coming up, you see lots of people that should be doing a regression or doing some plots, and they apply some complicated 10-layer deep learning algorithm, and who knows what to make of it, right? And it probably isn't necessary for most problems. For some problems, like the radiology problems we saw from Ron, they're very powerful, but that doesn't mean they work for every problem. So the suggestion, try simple methods first, move on to more complex methods, only if necessary. I'll always ask a postdoc I'm talking, did you try the simplest thing? If the answer is no, I'll say, go back and do that before you show me your complicated multi-step pipeline. Anyway, simplicity is something to be proud of, to try to aim towards. Okay, so now we'll talk about supervised learning and a couple of some sort of new advances from our lab. So this is, I don't know, I'm gonna go full screen here. Maybe this look better. Let's see. I should have done that. I'm afraid of breaking it. Dario, you seeing the slide that said the supervised learning paradigm?
Yep.
Slide 13. Yep. Okay.
Yeah, we're there.
This is the traditional supervised learning. We've got some regression data, I call it, training data, inputs, features, a target or outputs. We have a learning algorithm, and we apply the learning algorithm to the data. We get a model, using some model, and this model makes predictions for us on new data, and we output the prediction, right? So what's changed? Well, the big change really is in the data, I think, right? In the old days, an expert in some subfield would work for 10 years to collect good features in a certain disease, and people would then build small models using those features, and the statistician would fit the model with a selected set of features. Nowadays, that's pretty unusual. Nowadays, we can collect data much more easily with many, many different assays and machines and everything else. And we've got a large number of features. And now we need machine learning algorithms to try to sort through them and find the good ones. Okay. Main problems involve supervised learning. I'm on slide 14. We tend to sort of divide it conceptually into wide data and tall data. So the features are along the columns here of my data feature matrix, and the samples are along the rows. With wide data, you have large numbers of features, a lot more features than observations. And that happens, for example, in GWAS or gene expression studies, where the number of features is in the thousands or hundreds of thousands. Tall data, on the other hand, you've got a large number of observations and a small number of features. And the methods that tend to be useful for these are somewhat different. Random forest and gradient boosting, you would have all heard of it, are very useful for tall data, where you can drill down for interactions. For wide data, the real issue there is you want to do some kind of variable selection. Although, as Frank said, that's fraught with, that's a challenge, let me say, especially in terms of interpretability. I agree with him on that. But the lasso and elastic net are especially useful for wide data because they present a sparse model. They select through the features. Okay, so the lasso, which you very likely would have heard of, is basically a regression setup. You've got the features xij for the ith sample and jth feature with weights beta j, and this is the usual sum of squares from regression. And the lasso, the novelty is that you have the sparsity penalty. You say you have a budget on the total absolute value of the weights. Think of it as a budget. So I think of it this way: without this, we have our data. We can say I'm going to find the best fitting the weights to give us the best fit. But if, let's say, the number of features is larger than the number of observations, I can interpolate the data and fit perfectly. That's not a very useful solution. Rather, the lasso says, I'm going to give you a budget now on the total sum of the absolute values of the weights. And what that does is, if the budget is small enough, you get sparsity. The method will throw away features for which that are not predictive, give them weights 0, and put the non-zero weights on the features that are predictive, and do so with a budget which is determined by the data scientist. And this budget is determined by cross-validation. So that in one line is the lasso. It's a convex problem, which is very nice because it means that you can solve it. And I'll show you an example in a few minutes where we solve it for like 500,000 variables on a laptop in, actually, not quite true, maybe in a few hours at least. And then the sparsity is nice because it gives you feature selection. So this penalty is good for both computation and interpretation. So I'm now on slide 16. Our lab has written a package called GlimNet, about 10 years ago now, which fits the lasso and elastic net and other. And I want to just tell you today some of the new features in GlimNet, which is this is very widely used. We're proud to say it's 3.5 million downloads. That's as of 6 months ago now. It's in R and Fortran. It's also available in Python, psychedelics. So there's lots of software for lasso. GlimNet is, I think, as fast as any of them, and it's got a lot of extra features, which I'll tell you about. And it's been a team of people that have put this together. So the latest version, as of about 4 months ago, has got these kinds of features. Well, you can fit not just the Gaussian model, like the squared error I just showed you, but binomial, multinomial, Poisson. You can also define your own families in the GLM framework. And you can fit that now. There's group lasso for multi-response Gaussian. You can work with sparse matrices, which, for example, with SNP data is very important because the matrices are huge, but they're mostly zeros. You can do feature filtering within cross-validation. And now, most recently, we have a full treatment of survival analysis, just like the Cox pH in R. So there's the start-stop construction, time-dependent covariates, interval censoring is all there now. So we're trying to get a lot of the functionality that's in Cox pH, the very popular R package. It's now, not all of the functionality, but a lot of it is now in GlimNet. So that's very recent. So, how are we doing for time? Guess I'm fine. Okay. So I want to tell you about two things. One is called SNPNet. And this is basically extending the lasso and elastic net to GWAS, genome-wide association studies. So what's the problem? Well, with current software, for example, GlimNet or most lasso solvers that we know of, you can't apply it to GWAS size data, which means like 500,000 patients and 800,000 SNPs, for example. So we've developed a new approach using the idea of strong screening rules. This is work with PhD students at Stanford, Trevor Hasty, my colleague especially, and Manny Rivas, who's a genetic epidemiologist at Stanford. And there's a paper here, and there's a package called SNPNet, which you can use right now if you want. So what does it do? So with the high level idea is that as you run the lasso, you have a regularization parameter. The budget, or another way of putting it is in Lagrangian form, you start with a very sparse model. And then this is how GlimNet works. You go along, you start with a very large lambda, sparse model. Then you move along, and as lambda gets smaller, the budget gets bigger, and you allow more and more variables in. And that's how GlimNet works. It's a convex problem, but it's solved sort of along a path of regularization parameters. Well, the general idea of these strong rules is that having solved the problem at one lambda, say, and it enters maybe 5 variables, you can make a very good guess as to what the variables that are going to come in at the next lambda, a little smaller. Okay? The way we do that is called strong rules. Now, this is not an approximation. We're solving the convex problem exactly using the so-called KKT conditions, but we do so in a way that is much, much faster than the usual convex solver, because you can anticipate that, again, which variables are going to come in. You check the KKT conditions and you solve the problem exactly, far faster than you can solve with other solvers. So looking at the time, I'm not going to go into details of that except to flip to an example, which is from UK Biobank, 500,000 British adults, 805,000 SNPs. Whoops. And there's hundreds of phenotypes. That's the outcome from Biobank. We looked, for example, at a British subset of 337,000 patients, again, with 800,000 SNPs. So this is a big data set, right? This is too big to even put into memory in a laptop, or you need a pretty big computer just to get it into memory. Forget about trying to solve the lasso. We divide the data up into training, validation, and test set. The computation took a few hours on a 16-core machine. So this is not a huge supercomputer, but something which a lot of data centers will have. Again, we can't even attempt to solve this in GlimNet or most other L1 solvers. again, it's called the SNPnet package that's available on GitHub. If you're working in this area, you want to try it. Then we get a result like this. We get a result like these are this is the R squared as a function of lambda. You see the training and test are the black and red. And the others were the relaxed lasso, which in this case doesn't help. But the main point is here's the red curve. We can track the test set R squared, and it seems to be leveling off at around 50,000 SNPs. Of course, it's lower than the trace set. So you can do this kind of analysis, and you can do all the usual things that you do with supervised learning on these enormous data sets by again exploiting the sparsity of the problem and a strategy for dealing with it. Okay, so deep learning, the elephant in the room. We'll eat the lasso and other statistical models. Well, what is deep learning? Well, as Frank said, it's basically neural networks. Although the deep part really refers to the fact that now there's multiple hidden layers. Okay? In this picture, there's only one, but the deep part now, what's people using more and more often are multi-hidden layer neural networks. Sometimes with 50 or 100 layers. Okay. So what's changed since the invention of neural networks? In the 80s, they were invented, well, sort of more formally, and then they died down for many years. What's happened? Well, what's happened to lead to their success now, I think, is bigger training sets and faster computations. Many clever tricks have come up with convolution filters, stochastic gradient descent, and again, the use of multiple layers, which really only came out in the 2000s. And I was actually Jeff Hinton's colleague. Jeff Hinton is one of the grandfathers of neural networks. I was in Toronto for 13 years as a professor, and I knew Jeff very well. And I didn't appreciate the potential of neural networks at all. So I missed that boat. But I mean, I think I'm not alone. They're far ahead of their time computationally, and now they've really been shown to be useful in many problems. So why are they so useful? Why are they so powerful? Well, they're not one model, but they're customizable, right? Like we saw in Ron's talk, there's convolution filters you can use you can convolution filters, pooling. There's lots of ways you can take things you know about the problem and express that in a neural network system. Weight sharing, it's recurrent neural network. So there's a lot of very interesting tools which allow you to customize your model to the problem at hand. But does this mean the end of sort of all statistical methods? Will this apply neural network, deep nets all the time? Well, no. And hopefully I'm not just trying to protect my own territory. But in my experience and the experience of a lot of other people is that they're not a good approach when the number of observations is typically less than the number of features, and or the signal to noise ratio is very low, or interpretability is important. So they're most useful when the features have some spatial or temporal organization, like in the images we saw from Ron, time series, speech recognition. There's one-dimensional signals where one can exploit the structure, the time ordering in a strategic way. So this is really where they come to the fore. But sort of for generic data where their features aren't related in a spatial or temporal way, and or the signal to noise ratio is low, they tend not to work very well, and basically they're too complicated when you can do just as well with a simpler method. Okay, so having complained about neural networks, I'm going to say, well, if you can't beat them, you can join them. So we have made one contribution to neural networks, and that's called feature sparse neural networks, which I'm not going to describe due to lack of time, but I'm going to show you a little video, which hopefully will work, and will explain what our method is. So let me see if I can. This is a joint work with my graduate student, Ismail Lemhardi, and postdoc Feng Wang. So this is just 2 minutes long, but it's kind of fun, and it shows you what the idea is.
Wow, my neural net for diagnosing cancer has 95% test set accuracy. My boss would be very happy.
Hi, Ismail. How is it going?
Hey, boss. It's going great. My new neural net has 95% test set accuracy.
Great. So how does it work?
It works like this. There are connections from each feature to the hidden layer.
But which features are important for this task? our medical collaborators will ask about that. It won't be very useful unless you can answer that question. So let me know when you have an answer.
Maybe I should apply a lasso penalty to reduce the number of edges. Gee, that really didn't simplify anything. Aha, I have an idea. I'll add skip layer connections and use their weights as upper bounds for the hidden layer weights. That way, I can encourage the learning algorithm to eliminate all of the weights for a given feature. Nice. This has reduced the number of features from 5 down to just 2. I can't wait to show my boss.
Hey, Ismail, you wanted to see me?
Yes, I have simplified the network considerably. Now, it uses just 2 features, and there is no real loss of performance.
Fantastic, good work. I can't wait to show this to our collaborators. To say thanks, I'm giving you Saturday off, but I still need you to come in on Sunday.
Okay. So that's the idea of lasso net, and I'm not going to go into any detail. Well, let me just one detail. Oops. So the idea which you saw in the video is basically we have it's a res net, which actually Ron mentioned as well. There's a residual or skip layer, and we put a constraint. There's one idea. We put a constraint to say that the hidden layer weights are bounded above by the skip layer weights. So just like the lasso, you get a whole path of solutions, but now in a neural network, but it selects the features using an L1 penalty. So, but there's many, many more details, and you can apply this to any neural network, multiple layers, to any deep net. And there's a paper and there's software in Python we have, and I'm hoping people who apply neural networks in real problems, like for example, Ron, the previous speaker, will find this useful. So that's a sort of a marriage of lasso and sparsity with neural networks. So I won't talk about this because there's no time, but I am pursuing this as well with the collaborators in Toronto. We're looking at head and neck cancer from MRI images and clinical features. So we have lots of collaborations trying to see whether this can work in serious problems. So good timing. I'm just about to end. This is the last slide. So this is a plug for the second edition of our book with Gareth James, Daniela Witten, Trevor Hasty, myself. And we've added, this is Introduction to Statistical Learning, we've added new chapters on deep learning, survival analysis, and multiple testing. But I don't mind giving the plug because it's available for free at this website. The PDF is available for free. And we've also just completed, we have an online course, which we've had for about 8 years now, which covers this book. And now we've recorded some new lectures to cover the new topics. And that again is also a free course. It's an online course. Here's the URL for that. You just look, it's an edX. So if you just did a Google search for edX statistical learning, you'll find this course. And it includes new interviews with David Cox, Jeff Hinton, and Yoav Benjamini corresponding to each of these topics. And I mentioned, of course, that David Cox died about 5 days ago now, and which made me very sad. I knew David quite well since I was a graduate student, and he was one of my inspirations. And he was very kind at the age of 97 to give us to do a half-hour interview with Trevor and I over Zoom, and it was very interesting and entertaining. So if you want to sign up for the course, again, it's free. You can listen to the interview with David and also with Jeff Hinton and Yoav Benjamini. And that's it.
Thanks, Rob. Amazing talk, amazing presentation. And we can start maybe immediately the discussion, and if Frank and Ron would also switch on the video, so we'll simulate some sort of true roundtable. We have a very diverse set of questions because we have students from biostatistics, but also students from the medical school in general. And so I'm trying to put together some general lines, and I think that the one first that maybe is worth to go a little bit ask to all of you is about the concept of validation, because some people talk about the validation with different type of data, omics data, imaging, or more traditional features. So the question is that changing the validation process as you change the data or the field of application, changing the concept of validation? For instance, I have seen Ron use a kind of a heuristic way of thinking of validation, like matching human classification with machine automated classification. In other fields is more kind of a in a statistical sense. So what do you think? I may start with Ron and then Rob and Frank.
Right. Well, you know, in the AI field in radiology, which we used to call computer-aided diagnosis, but it got a name change, just as Frank said. The neural networks got a name change. We used to do leave one out cross-validation, and then we moved to you know, n-fold cross-validation, and now people seem to be focusing on separate training, validation, and test sets. So the types of validate and the journals are generally requiring, in addition to an internal validation, also an external validation, which gets at some of the interesting points Frank made in his talk about the issue of what happens if the external validation doesn't show good generalizability. So that's kind of where we are from the practical point of view. I'd be interested to hear what the others have to say.
Yeah, I mean, I'd agree that, well, the kind of validation you do, first of all, this probably depends on how much data you have. But if you can afford to have, you know, a separate validation set and a large test set, then I think it's ideal to use a validation set. If the data is not large enough, then cross-validation's a good surrogate for validation. I do agree that, I actually agree with the journals that although cross-validation in theory gives an unbiased estimate of test error, it's in theory only because, you know, if you do cross-validation once, you can pretty much read off the minimum error as a reasonable estimate of test error, although it's a little too low. But in fact, what people do in practice, all of us, right, is you do cross-validation on your model, you're not happy with it, you change the model, you get some more, you know, it's an iterative process. So it's very important to have a final test set sort of as a real objective measure of how well your modeling pipeline has gone. So I agree with the journals in that having a separate test set is a very good idea. And even beyond that is, right, you may have a separate test set that does well, and then someone else across the country tries your method and, hey, it doesn't work like you guys said it was going to work. Well, it's not like you did anything wrong, but, you know, patient populations are different and the medical practice is different. So validation is really important. It's a multiple stage thing in the whole scientific process, right? You know, we do validation, try to do the best we can, and have other groups try to validate. Eventually, we might converge on a good thing. Yeah. I don't know if Frank wants to add anything.
I'll just stir up the pot a little bit. I really love resampling much more than holdout samples. In the case where N is greater than P, because I think the bootstrap can understate overfitting if N is much lower than P. I just don't like to hold out data if I can boost my sample size. And then I really like resimulation, where you simulate from your fit to generate new data, and then you see how you can recover the truth. So I like simulation often more than I like validation, just to study properties of methods. And you have to work hard to make the simulation realistic. And then the last point is, two other quick points. I think of external validation as something that involves data that were not available at publication time. Because if the data were available at publication time and you held it out, you're really lowering your information for your analysis just to be able to do a so-called unbiased validation. So my working definition of external validation is validation on data not available to the analyst at the time they wanted to finish the project. That's kind of a radical idea. And my last point is the methods that you validate, the indexes that you validate, get back to the points about, do you have spatial or high signal to noise ratio data where classification is more interesting. And so do you validate on the basis of classification accuracy? I think that the number of times when it's good to validate on classification error accuracy is fairly much in the minority compared to validating the accuracy of prediction. So I believe that most analyses are interested in analysis of tendencies, whereas a classifier is not an analysis of a tendency. It's analysis that's trying to make a decision like you really are able to do in visual pattern recognition. So validation to me, like in the example that Rob gave in the cartoon, the 94 and the 95, I'm not sure those are the best settings for classification accuracy to be what excites the guy in the cartoon.
I want to sort of maybe disagree a bit with what Frank said. So the first point about the bootstrap, I mean, I'm obviously a very big fan of the bootstrap. I mean, I wrote a book about it with Brad Efron, but it's no replacement for leaving data out. The problem with the bootstrap, right, is it's when the bootstrap samples have overlap with the original sample by definition. They're drawn from the original sample. So for the assessment of prediction error, they're overly optimistic, right? Because you're predicting. I know Frank knows this, but I just want to say it anyway for the students that you're predicting data points, some data points which you've already seen. So cross validation and validation to me is uniquely important because you actually separate the data from training and validation. So bootstrap.
Let me fuss about that a bit. So now my simulations are focused on the cases where n is much greater than p.
Okay.
But in those cases, I don't like to think of bias as the gold standard. I like to think of mean squared error as the gold standard. So if you do simulations to show the mean squared error of what you're estimating with the bootstrap, the mean squared error is typically much lower with the bootstrap than it is with holdout sample methods.
Well, hang on, it's lower. The question is, okay, so you have a procedure, and there's a true mean square error you'd like to estimate, which you get if you had a test set, which an infinite test set, which
is how I do the simulations.
Okay, but so the question is,
simulate an infinite test sample.
We're not going to simulation. I'm talking about bootstrap versus cross validate. Bootstrap will give you a lower mean square error, but it's a fiction, right? It's lower than the real one you're trying to estimate. It's biased.
No, I'm saying the mean squared error of the bootstrap.
Yeah.
In terms of the means, let's say that what you're estimating is ROC area.
Yeah.
The mean squared error of the ROC area with the bootstrap is typically lower than the mean squared error of the ROC area estimated with a holdout sample.
Well, I don't. The mean square error might be lower, but it'll also be biased downward. So I guess you're saying you don't care about the bias as much as the mean square error.
Right. If it's unstable, I want to penalize for the instability. So I don't care about bias. I care about the probability of being close to the truth.
Okay, well, I would say if you care about that, then do repeated cross validation. That'll lower the standard error.
Yeah, I like that too. Yeah.
Okay.
Yeah.
Okay, well, we don't want. Okay, let's back to Dario.
Yeah, well, we have I think we have two different type of questions, but they are somehow related. In general, not very technical, but one is maybe for Ron is, okay, all this artificial intelligence will replace the radiologist and the doctor, and on the other side will be all this machine learning stuff replacing statisticians?
All right, I guess I'll start with the radiologists. I don't really see these machine learning algorithms replacing radiologists, but I see them being used by the radiologists to either improve their efficiency or improve the quality of their diagnoses. So, for example, radiology reporting has traditionally been a very qualitative exercise where the radiologists look at the gross pattern information in the image and they report things like there's been a slight increase or there's been a moderate increase, or, you know, and the most quantitative they ever get is the lesion used to be 3 centimeters and now it's 3.5 centimeters. But I think in the future, you're going to start seeing radiology reports where there'll be a section with all sorts of machine learning generated numbers, like these are the volumes of all the different organs. This is the quality of the muscle. This is the amount of fat and so forth. And then population-based estimates. So for example, traditionally in bone mineral densitometry, you get some statistical assessment like a Z-score, a T-score, assessing for a particular patient of a particular gender and age. How do they compare to other people of the same gender and age? And I think we'll start to see that more frequently in the radiology reports, this sort of quantitative section. So that's not replacing the radiologist, that's doing something that the radiologists are not going to do because it's too time consuming to manually make these assessments. And computers are great at doing things like measuring an organ in 3D and then doing the simple multiplications and adds to determine how the person stands on some nomogram, relevant nomogram of according to that patient's gender, age, or maybe even race or ethnicity. There might be other categories as the analyses get to be more sophisticated. So just to summarize, I think these will be additive tools that will improve care.
Oh, thanks, Ron.
I'll let Frank tell us whether statisticians are going to be replaced by computers. I think I know the answer.
My answer would be that bad statisticians are going to be replaced by computers. Bad radiologists are going to be replaced by computers.
Okay, so we should our PhD students biostat that should switch to other majors like data science or something like that, or should we?
You know, Jeff Hinton actually was videoed saying that physicians should not become radiologists because the field would be gone in 5 years. So I think he's backtracked on that statement since, but you know how it is once something's recorded for posterity, it tends to keep coming up.
Wonderful. Okay, and I have a question that I think is mostly for Frank, but I would really love to have also the reaction from Rob and Ron about the experimental design, the clinical trial settings, which seems to be disappearing from all this field. We barely touched the issues today, and but still, I mean, clinical trials are. I mean, it's not just to say they are the gold standard. This is how we demonstrate efficacy of whatever we have to implement in the medical field. So, Frank, do you think it's machine learning idea, statistical learning idea, fitting also into the clinical trial setting?
I think, of course, the need for clinical trials is greater than ever, and that will not be changed by any of this technology. I think the place that machine learning is most likely to help is what Rob touched on with heterogeneity of treatment effect. But I'm not going to take that field seriously until I see somebody come up with a way to do a sample size calculation that shows you what sample size is needed to analyze heterogeneity of treatment effect whether it's with machine learning or not. And then the budgets of clinical trials are increased enough to achieve the sample size necessary to do reliable heterogeneity of treatment effect estimation. So there's a lot of people doing this, a lot of statisticians doing this without any examination of or validation of the heterogeneity of treatment effect signals.
I agree with Frank. Actually, I have a student working right now on this problem, trying to find subgroups of patients. You know, you run a clinical trial, say, and the average treatment effect is null, but you want to find subgroups of patients for which the treatment might help. We have what we think is a very clever method, but we find exactly as Frank is saying that in simulations, the power needed, the power is very low unless the sample size is huge and the heterogeneity is large. I mean, looking for a subgroup like that is looking for the interaction between some features and a treatment. And interactions notoriously require more data. So he's absolutely right. Lots of people are doing this, and a lot of it is, I think, futile because we don't have the power to find such subgroups. But that means hopeless, but it's, yeah.
That's a way better way to say it. And just to nail the coffin, the typical clinical trial has a sample size that's inadequate for estimating the average treatment effect.
Right.
So if it's inadequate for estimating the average, the situation that Rob just described is pretty dire.
Yeah. But it's something important to pursue it. And, you know, if the budgets permit, we tell us maybe these things can be done in the future, but most of the data sets we find archivally are just too small. And the effects you need to look for have to be huge before I can find them. So.
There's a data set I keep analyzing over and over with a 40,000 patient randomized trial. It's a beautiful study called Gusto-1, a heart attack study. And when you have enough data, I think you can really demonstrate there is no heterogeneity of treatment effect, because I've given every opportunity for heterogeneity to be found in that 40,000 patient data set. There is not a hint of heterogeneity.
Yeah.
Okay, thanks. I have another question coming from Eric, who is a PhD student, and he was also interested in the concept that Rob you introduced about the abstain category in classification. So can you go a little bit further on that, or is this category something which is it's something about somehow related to all the fuzzy classification systems?
Well, I can say more. So again, what the problem is, and I've had personal experience with this because I have a collaborator that a longtime collaborator. He uses a certain kind of mass spec to use to try to predict the presence of cancer, try to detect cancer from the spectrum. So basically, it's a high-dimensional classification problem where you have maybe 20,000 features and a few hundred patients, and there's two classes: healthy or cancer. Right. So we build classifiers. We built classifiers on this data using Lasso or other things, random forest, and they seem to predict well using cross-validation. But then the problem is, the challenge is now when we have a new patient coming in, their query vector, feature vector, is also 50,000 long. But if you think about it geometrically, their feature vector could be far away from the training set. For example, I mean, in real terms, this patient could be quite different than the patients you've seen in your training set, right? And you can run it through the classifier. The classifier says, I'm sure it's happy, it's class cancer with high probability too. It says with 0.9 probability, I predict this to be cancer. But in fact, it can be totally wrong because the model is extrapolating, right? And you have to imagine extrapolation in 50,000 dimensional space is really easy, right? It's not easy, it's hard. It happens a lot, right? Because the data is so sparse in the space that almost everything's an extrapolation. So in that situation, if you want to use such a system in actual clinical practice, you want the system to be able to say, sometimes when the query comes in, to say, I'm not going to make a prediction. I don't have enough information. I want to abstain. This patient is too different from what I've seen in my training set to make a prediction. So it's a real problem. And there's no easy solution. It's because, again, you're having to detect when you're extrapolating. And in 50,000 dimensional space, maybe only the first 100 features are important. So it's sort of a chicken and egg problem, right? The problem's actually lower dimensional, but you don't know that. You're trying to fit a model, and extrapolation is a problem. So I don't know if I explained that, but to the questioner, but it's a real problem that you're faced anytime you're using a classifier to do real-time decision-making with a large number of features.
Yeah, thanks, Rob. Frank, Ron, do you want to add something to this, or?
I would, if I could just add something brief. Sorry, Frank. I'll be brief. We've been trying to add uncertainty estimation to our predictions. I think that's something that the radiology AI field has been a bit behind the curve on. So, for example, for segmentation, for classification, just reporting error metrics. We frequently make a prediction of a single classification. We don't mention other classification labels that might be equally plausible within the probability estimation. And I think that you could imagine a scenario where none of the labels achieve sufficient likelihood that you would abstain. You would get a null set for a label for classification.
And just to be clear, and this problem is hard. Uncertainty measure is important, but you can't, for example, use the bootstrap to solve this, right? Because imagine you draw bootstrap samples, they all come from your training set. So you have the same problem. The bootstrap can't detect extrapolation because it's just seeing the data you give it. So this problem is sort of one order harder than just internal resampling. So I guess I'm saying I don't know how to get uncertainty measures to solve this problem. Uncertainty measures are really important, but they're not there yet for extrapolation problem. Yeah.
If you were completely free of the extrapolation problem and you had just a simple situation and it was kind of low dimensional, classification is at its best when you could have done probability estimation, but all the probabilities are close to 0 or 1. So you don't have any gray zones. But if not all the probabilities are close to 0 or 1, then to me, the probability estimate is the best index of uncertainty because it's built in with its own uncertainty. So if you had a probability of 0.51, if you wanted to assume disease is present, it was a very tough call and you're on thin ice.
But probabilities don't solve this. Again, I can show examples where the classifier says, probably 0.9, you've got cancer. And it's completely wrong because it's extrapolated.
Yeah, I was talking about the situation when that didn't happen.
That didn't happen. Okay. Yeah.
About the simple situation, no extrapolation.
I agree. Right. So yeah, the probabilities are really important. Yeah.
Wonderful. I got a question from Daniele, and I think it's a very interesting topic in a broader sense. When you do cross-validation, internal, external, and so on, if you have structure in the data, is it worth to keep the structure? Let's say, put some center in, put some center out, or maybe just pull them together and do cross-validation, mixing the information, losing the structure in the data.
Well, what's an example of structure? There's maybe time.
Like hospital. I have 20 hospitals, and instead of having using a cross-validation based on the individual patients, you do cross-validation on the clusters. Is that something you gain or you lose?
Like I say, I think if there's big between hospital differences and you mix and you don't respect the hospitals in your cross-validation, so that there's a given hospitals both in the training part and the test validation part, I think that's problematic because let's see, let me think this through. So I guess it depends on whether you want an assessment of prediction error that's hospital aware or not, right? You can imagine both.
Yeah.
So if you want a hospital aware prediction error, so that you would know then I guess it's fine to mix the hospitals between folds. But if you want something which is hospital independent, then I think you should keep them separate. I don't know if the other speakers agree. Well,
what do you think about this question? Is it worthwhile to make the predictions hospital specific? More worried about that than the validation hospital specific. In other words, if you have random effects for hospitals, you'll be able to recognize the heterogeneity, and you'll also be more able to make predictions for a hospital that's not in your data.
Yeah, I'd agree with that. If you have enough data, then do fit separate hospital models, and you can learn about the heterogeneity. Yeah.
But I was suggesting a unified model with, say, random effect for
Oh,
hospital.
Yeah. Oh, yeah. That's a good idea too.
Wonderful. I have another couple of questions, which I translate in something which may sound naive, but is, of course, coming to the mind of each one of the practitioners and the medical doctors that are dealing with this in our school. I mean, if you have a prediction or a classification problem, which will be the machine learning method that you will suggest to start from? Simple regression, as long as data allow that, or random forest, or lasso, or neural networks, or whatever. What will be your first choice?
I made a push in my talk for starting with simpler methods, so I'll just restate that, right? Start with logistic regression, say. Well, first of all, start with some plots.
That's okay. That I agree with that. We also showed that in some.
Start with simpler methods for sure. Yeah, and then again, don't try just one thing. Of course, you should try lots of things and using validation, cross-validation to compare. But start with simple things that might be all you ever need for some problems, for many problems.
Frank, do you want to add anything?
Just to throw out one other angle on this is sometimes you use hybrid methods that have the possibility of being simple but may not be. So an example would be you have all of the features in a general linear model, and then you use a penalty for all of the two-way interactions of those features, and you cross-validate to solve for that penalty, but you don't penalize the main effects. That's kind of a compromise model, I think.
In this respect, about simple models, we have a question from Corrado, who will be a panelist dating tomorrow. What about your feelings about combining information from structured and unstructured data? I mean, things comes much more difficult. I think Rob was telling us about the idea of a very nice and clean data sets coming from our clinicians. And we were used to do that. Now we have a lot of data which are not cleaned before the analysis, so it may not be as clean as it was. So how do you, what is your feelings about the perspective of using such data? And this is for all of the three speakers.
Well, I think, for example, from clinical notes, which are unstructured, if that's, if that can be a very useful source of information. And it's challenging. You have to use things like NLP to extract the information to make it more structured, but you certainly want to use unstructured data if it's potentially useful.
Ron, did you combine clinical notes, radiology, or improving classification, imaging?
Well, we have a collaboration with the National Library of Medicine where there are experts on NLP, and we have been working on that and using NLP on the clinical notes. The clinical notes are so variable in their structure and dictation styles of the different radiologists that there are quite a number of issues that come into play. That's why when you have things like structured reporting or some sort of what they call reporting and data systems, which are increasing in number in radiology, you can get more accurate labeling from NLP algorithms. But there's a lot of noise in these NLP generated labels. And yet when you have very large data sets like 100,000 reports or 250,000 reports, you can still do some interesting things. So for example, for the chest radiograph data set, we put out a data set with, I think it was about 120,000 chest radiographs. We generated the labels using NLP, and we call it the NIH Chest X-ray 8 data set because there were 8 different labels that were extracted from the NLP. But there are a lot of problems with that because in radiology, it's not just the presence of an abnormality that's important, it's also its severity and location. And you have noise in figuring out the severity and the location. Another thing that's common in radiology reports is qualitative uncertainty estimation. So there are different ways that radiologists express uncertainty in a finding, and uncertainty is a big issue because the scanning modalities are frequently very good at telling you where an abnormality is and what its shape and size are, but it's not always good at telling you what it is. So you get uncertainty in the report that then propagates through these different AI studies. So it's a really interesting area.
Thanks, Frank. Do you want to add something?
Yeah, there's some nice work being done by Brian Shepard and others on data audits, where you might have 10,000 patients, but you audit 300 of those, and you get a panel of experts to make the gold standard determination, and then you estimate the probability that the natural language processing was correct in one direction, and what's the probability it's correct in another direction. And you use those audit checks on the data and what the uncertainties were in a formal way in your final analysis.
Tomorrow's session will be focusing on NLP with 2 excellent speakers. So we will see tomorrow how things goes. I have a final question, which is very broad, and I will also love to have Professor Angelini joining us because it's really on the foundation. Is we are teaching statistics in the medical school. I have seen, Frank, you already answer on the Q&A panel, but basically the idea is shall we change the way we teach statistics in the medical school for medical students? Shall we introduce new concepts, new way of learning? What isn't that we will produce a generation of medical doctors that will know all concepts like T-student and T-test and so on, but they will miss some mechanisms that are used nowadays? I will see Frank and Rob and Ron.
Well, I think when you teach study design, they never forget it. When you teach an analytical method, they'll forget it as soon as they're out the door. So that's why I think teaching study design and teaching measurement, there's a whole theory of measurement, and David Hand has an amazing book called Measurement. Teaching those 2 things those skills will last a lifetime, and they'll matter in every context.
I guess I would also add to that, teaching critical thinking, right? I mean, a lot of these people may not ever go and do the actual analysis themselves, but they're going to be reading papers, and they have to be able to critique them, right? So they have to be able to judge them, find the important aspects, find the shortcomings. So critical thinking about statistics and data science is really important to teach, I think, as well.
I agree. Do you want to, as a medical doctors, do you want to put your perspective?
Well, I think it's so important to engage with the statisticians when developing one's study design. I've learned so much from working with statisticians. I think there's so many open questions too. We've heard about some of the issues, for example, sample size. How do you determine sample size appropriately for an imaging related deep learning project? I'd love to see Frank or Rob help me figure out how many images I need to do a machine learning experiment. We're completely clueless in that regard. And so you're in this situation where everybody just thinks, oh, the bigger, the better. And if you have a sample of only 100 images, well, that's not enough. You know, this paper's rejected because it's only 100, but on the basis of what? I mean, does anybody really understand the variability? So I think what I would say is that understanding the role of uncertainty in analyses is critically important. There's really little understanding in my field about how important it is to not only report an estimate of the mean, but also an estimate of the uncertainty in the mean. And there's so many papers in our community and other communities where tables of data just show the mean of whatever parameter, and there are no error bars, and there are no statistical tests being used, and everybody's just trying to increase the value of the mean by 1 percentage point in order to say that their method is better. And I think we need a lot of help from people like Rob and Frank to reduce the noise in our field, which are leading to a lot of wasted effort and potentially not helping patients the way we would like to. That's the whole point of what we're doing, trying to help patients.
I'd like to second that motion a lot. I mean, uncertainty measures really were badly needed. And one example of that, I've been working in the COVID field and forecasting part-time. And probably the best known website for this is IHME at University of Washington, which has a very popular website for forecasting COVID cases. And they present prediction intervals which are terribly calibrated. They claim they're 10%, 90%. And if you actually look at their achieved coverage post hoc, which people have done, they're more like their coverage is more like 50%, right? And when this is pointed out to this group, they basically ignore it. They say put these intervals up, which are completely they're far narrower than they should be, and they're communicating a level of certainty that just isn't there, right? I mean, the fact of the matter is most groups, almost every group that tries to forecast COVID can't do it. It's just too hard, right? But of course, everybody wants us to tell you the answer. When they look, it's going to go down tomorrow. But we don't know. That's the answer most of the time. But that's not satisfactory somehow to politicians or society. But it's really important to understand that uncertainty measures, especially when they're so large as to make the prediction useless. Because people use these forecasts to make policy, and they need to know what the level of scientific evidence is. So I really agree with that strongly. Uncertainty.
Yeah, we can go back to the first slides of today's where Frank was talking about the selection of researchers, not only selection of features. Mm-hmm. And if you go back to the first couple of months of the epidemics, like first wave, I counted about 200 clinical predictive models published in the 3 months after the starting of the epidemic. So and few of them were even validated, or I wouldn't say post-hoc coverage or something like that. It was just produced. And so this is something that we're trying to put in criticism for our PhD students. And I don't know, Annalisa, if you want to add something.
Yes, if I, as a medical doctor, I was thinking I like very much this aspect of critical thinking that we should teach. And also because we must stress for the students the need of the quality of the data collected, because I think this is also an important aspect before going to the statistical elaboration and then interpretation of the results. Mm-hmm. Because sometimes we are missing this aspect of the quality of the pre-analytical data. And instead, I think is a very important aspect, a critical aspect also, the quality of the data and how, for example, artifacts or problems in imaging can interfere and with the good interpretation of the data. So I think this is an important aspect. Maybe being a pathologist, one of the major aspects is that with digital pathology, for example, we are facing the problem of artifacts and how to overcome this aspect in elaboration and artificial intelligence. I hope that on Thursday we will come back to discuss this aspect. But I think the quality of the data are an important point that we should teach to the students.
Yeah, I agree. And this is part of the job of the medical doctor and biologist working in our research labs and so on. Okay, I think we are slightly over the time. I think we mostly touch all the points raised in the Q&A section, and if not, we will forward the question, try to provide an answer afterwards. I just want to close really expressing my gratitude to all the three speakers, excellent as expected, but also very friendly for accepting to give this this presentation in a time zone which is absolutely not convenient for them. And so thanks again in order, Frank, On and Rob, and I hope we all stay in touch also for the future development of our PhD program in Padova. And so thanks a lot and see you sooner than later.
Thank you very much. Thanks for organizing.
Thank you very much.
Good job. Super.
It was a
stuff.
great pleasure. Yeah.
Really enjoyed it.
Thank you.
Thank you.
Thank you.
Many thanks. Bye.
Thank
See you tomorrow afternoon.
Bye.